Someone has very rightly said that a small good thought in the morning can change your whole day. It means that once you get up in the morning, then your thought process for the whole day should be positive. You should not get up from your bed with a negative thought that, oh, again a whole day is there, I have to study, I have to do this, that. No. Every morning you should have a positive thought. Get up from the bed recognizing the God and thanking God that yes, today also He has given you another day to do and to give your best. If you get up early in the morning with some positive thoughts, then I assure you that your whole day is going to stay positive, full of energy and full of enthusiasm. So with this simple thought of the day, I, Ayushi Agarwal, is going to welcome you on this amazing platform of Physics Wallah, where today we are going to learn the next very important chapter of class 11th, that is Cell, the Unit of Life. Now, this chapter is not new to anybody. Since childhood, we have been hearing about cell, cell and cell. Some teachers might be telling you this is cell, some might be telling you that is cell. So now today we are going to gather all our childhood memories and childhood information regarding cell, and we are going to start with this new and very important topic. So the chapter is going to start with a very simple definition of what is cell. So how can you define? Just go in your childhood and try to recollect the answer. You will get a very simple answer that cell is the basic structural and functional unit of life. All living organisms without any exception are made up of cells or product of cells. Yes or no? Anything which is living has to be made up of cell. Any structure which is less than a cell is not considered as living. So many thoughts arises when I talk about what is cell. So the definition part I have already told you. Cell is the basic structural and functional unit of life. Basic structural and functional unit of life. Everybody agrees to it? All living organisms are made up of cells, maybe single cell or maybe multi-cells. If the body is made up of one cell, then such organism is considered as unicellular organism. And that unicellular organism is also capable of performing all those metabolic functions which a multi-celled organism can. That means cell is actually the basic unit. If any object is made up of even one cell, then that body has an independent existence without any doubt. So all organisms are composed of cells, right? Some may be unicellular, some may be multicellular. Unicellular organisms are also capable of independent existence and are capable of performing all the essential functions of life. If I talk about bacteria, amoeba, euglena, all these are single celled, but still they have a proper body organization and they have all those structures which are responsible for existing a sustainable life. Correct? Moving ahead. Anything that is less than a cell is not considered as living. So we can simply say that cell is the structural and functional unit of life. Correct? The term cell was coined by Robert Hooke. He was the scientist who was studying on what? He was studying on the cork. He cut the thin slice of a cork of a bottle and placed under the microscope, where he observed some compartment-like structures. These compartment-like structures, these honeycomb-like structures, honeycomb-like structures by Robert Hooke was termed as cella. Cella itself means cell. So Robert Hooke was the scientist who coined the term cell. Actually, Robert Hooke was observing the cell wall. Thus, by observing cell wall only, he considered it as a cell, and there the terminology came into existence. However, Robert Hooke discovered the dead cell because it's a cork, and cork is a dead part. Correct? So, if question comes who coined the term cell, then your answer will be Robert Hooke. But if the question comes who was the first one to see a living cell, then your answer will be Antony von Leeuwenhoek. So moving ahead, that who is the scientist who observed the first living cell was Antony von Leeuwenhoek. He was actually working on mud water. He took a mud water, right, and placed it under microscope, where he observed some moving organisms which he called as animalcules. And those animalcules were actually rich in spermatozoa, some other unicellular organisms like bacteria and all. So from there, cell term was coined by Robert Hooke. However, he observed a dead cell. To be specific, he observed the cell wall. And the first living cell was described by Antony von Leeuwenhoek. Now, like I say that my body, whole my body is under the control of brain. Whatever activities I am performing, talking, singing, dancing, all those activities overall they are controlled by my brain. If brain stops functioning, if brain stops sending the signal, my body coordination is going to get disturbed. So like my overall body is controlled by brain, similarly each cell has its own brain which is called as nucleus, that encloses the genetic material. So nucleus is actually brain of the cell which is responsible for controlling all the metabolic activities which are taking place inside the cell. And this nucleus was discovered by Robert Brown. Clear to everyone? So now you have learnt three names of the scientist: Robert Hooke, Antony von Leeuwenhoek, and third is Robert Brown. I hope you are going to remember all the three names of the scientists along with their work. Now moving ahead is cell theory. Very simple, again a topic of 6th and 7th standard. So now let's see what is cell theory. There were two scientists, one a German botanist and second a British zoologist. German botanist was Matthias Schleiden, whereas British zoologist was Theodor Schwann. These two people in two different years, living in two different countries, working on two different cells, proposed some points and later came to know that their points are somewhere similar. So their postulates, their points were joined together to frame the cell theory. So now let's see what are those points explained by Matthias Schleiden and Theodor Schwann. So Matthias Schleiden, who is a German botanist in the year 1838. Being a botanist, he was studying on plant cell and said that all plants, their body is made up of cells. And cells in return, they join together to form the tissues inside the plant body. So what Matthias Schleiden stated? Matthias Schleiden in the year 1838, he's a German botanist. He stated that plants, their body of plants are made up of cells which joins together to form tissues. So in short, you can say Schleiden wanted to say that for plants, the basic unit is cell. Correct? Now, talking about the next scientist in the year 1839. Can you see? It's a 1 year gap. And this fellow, Theodor Schwann, is a British zoologist. He was studying on animal cell, and he said that animal cells, they are surrounded by a delimiting thin membrane called plasma membrane, whereas plant cells have one more additional layer called cell wall, and presence of cell wall is a unique character of plants. Along with these two statements, Schwann also stated that be it plants or be it animals, all living organisms, they are made up of cells or the product of cells. So in short, you can say that Schleiden and Schwann, they both wanted to convey one thing, that all living organisms, irrespective of being plants or animals, are made up of cells or product of cells. So can you see there's a common point between two different people proposing two different theory in two different time? So their postulates were joined together to formulate cell theory. Correct? So what are the points of cell theory? What cell theory says? Number 1 point, that cell is the basic structural and functional unit of body. First point. Second postulate is what? All living organisms are made up of cells or product of cells. Product of cells means tissues, organ system. So all living organisms, correct, are made of cells or product of cells. Correct? Third point, which is also included in cell theory, states that whatever metabolic activity a living organism is performing is sum total interaction of their cells. Right now, if talking, I'm walking, I'm eating. So these are the metabolic activities which I am performing. And this metabolic activities is because the cells which are making my body, they are interacting with each other. And it is the result of their interaction. So third postulate says that whatsoever the metabolic activities performed by living organism, correct, is the result of their cellular interaction. Oh, just a second. Living organism is the result of their cellular interaction. Clear? I hope all the 3 points of cell theory are clear. Now, this cell theory given by these two people seems to be little bit incomplete. How? Because this theory is telling everything about the cell, importance of cell, but this theory is not telling that from where are the cells arising. Thus, this cell theory later was modified in the year 1855 by a Swedish scientist called Rudolf Virchow. Rudolf Virchow, a Swedish scientist, he gave a statement called Omnis Cellula e Cellulae. This statement says that all the cells, they arises from their pre-existing cell. That means if there's a parent cell, then this parent cell is going to grow in size. A time will come, it will undergo division to produce new daughter cells. Now, these two daughter cells will also grow in size. A time will come, they will divide to produce new sets of cell. That means all the cells, they arises from their pre-existing cell. Cell shows lineage. Correct? So this statement, omnis cellula e cellulae, modifies the cell theory and completes the cell theory. Is it clear to everybody that what is cell and what is cell theory? Now, moving ahead with the chapter, that is overview of a cell. Now you know what is cell, you know what is cell theory. Now let's see how does a cell appears to be in terms of shape, size and function. So these all things we are going to study under overview of a cell. If I talk about shapes and sizes, then cell shows great diversity in terms of shapes and sizes. There are numerous shapes of the cell. Some cells can be cuboidal, columnar, and some can be elongated, some can be disc-like. So many shapes are present of cells. Cells are of no definite shape. You cannot say that, ki ma'am always draws cell in circular manner, so cell always has to be like this. No, cell can be elongated also. Cell can be columnar also. So cell has different shapes, as well as they also vary in terms of sizes. The smallest cell is of mycoplasma, which is about 0.01 to 0.02 microns. And the cell size can range up to, like for example, the longest cell is of neuron. The RBC diameter, the size of an RBC is about 7 micrometers. So you can see that in terms of shapes and sizes, the cell shows so much diversity. It has different shapes and range of size of the cell also ranges from 0.03 to 7 microns, even longer than that. Right? So what are you going to write in overview of the cell? What shall be your first point? Yes. So in overview of a cell, your first point is going to be cell shows great diversity. Cell shows great diversity in terms of shapes and sizes. Correct? This is the number one point. Like for example, shapes of the cell, they can be variable. Some cells may be columnar, some may be cuboidal, some may be polygonal, some may be elongated like tracheids. So shapes are variable. If I talk about size also, then size of the cell is also highly variable. The smallest cell, which is mycoplasma, also known as PPLO, pleuropneumonia-like organism. It's a smallest bacteria whose size may range from 0.1 to 0.3 microns. Right? So this is the smallest size of any cell. Now, cells can be as long as RBC, human RBC. The diameter, the size ranges up to 7 micrometers. The longest cell is of neuron. Yes or no? These information you should know. Question can be asked in your NEET examination. The largest cell is of ostrich's egg. Correct? So please students, do remember all these information given in your NCERT so that if questions are asked from this section, then you don't make any mistake and you secure all 4 marks. Now, one more point I would like to add in this. When I say cell shows diversity in terms of shapes and sizes, then have I mentioned about function? No. Now, function. How will you decide that which cell is going to perform which function? So a simple answer to this statement is that shape of the cell decides its function. What have I said? Focus everybody. I have said that the shape of the cell decides the function it performs. What is this? So now let's try to understand this statement, which is very, very important in terms of an example. Now, I'll take an example of a trachea. So what is the shape of a trachea? The trachea, which is made up of long elongated cells called fibers. Now, tracheae, the individual cell of a trachea is long elongated pipe-like. Why these cells are not spherical? Why these cells are not triangular? Why these cells are not polygonal? And why are these long elongated? They are long elongated pipe-like because their function is conduction. They are responsible for conducting water and mineral from the roots to the leaves. And the best shape for this function is long pipe-like. Have you ever seen any pipe in your home which is spherical, which is triangular? No. All the pipe, they are long, elongated, cylindrical in shape. Why? Because when pipes are of this shape, then conduction becomes easy and convenient. So, tracheae, they are made up of elongated cells only because their function is conduction. So now you must have understood that why shapes are necessary for deciding the function which the cell has to perform with this simple example of tracheae. So tracheae, they are long elongated in shape in order to make conduction of water more simple and easy. So shall we write this point? Like for example, tracheae are long elongated for what? For efficient conduction of water. Correct? Okay, very good. So this much you have to do for overview of a cell. One more point I'm going to add over here, and that point is that now we can divide cell on the basis of their genetic material, on the basis of their nucleus found inside, into two broad categories. But before doing that, you should know all the cells, they encloses a fluid inside it which is responsible, which is the main arena for all the cellular activity, and it is known as cytoplasm. So next point which you have to know and which you should note down is that cytoplasm is the fluid or is the main arena where all cellular activities takes place. And it is found in all the types of cells, cellular activities takes place. This is very important. This line is given in NCERT and has been also asked in NEET. So this line becomes very important. Question will directly come. The dash is the main arena of all cellular activities. Some options will be framed, but your answer should always be cytoplasm. Now, I was telling you about that cells on the basis of the nucleus, on the basis of their genetic material, that is nucleus, they can be broadly in two types. We can broadly divide cell into two categories on the basis of their nature of genetic material. That is prokaryotic cell and eukaryotic cell. Is it new terms? No. These terms we have heard again so many times. It's just a quick revision. Pro means what? My dear students, pro means primitive, old, not new. And Karyo means nucleus. So those cells, those primitive cells who does not have a well-defined genetic material are considered as prokaryotic cell. Contrary to them are eukaryotic cells. Eu means true, advance. And karyo again means nucleus. So those cells who have a well-defined genetic material and their advanced cells are considered as eukaryotic cell. Now when I say advanced or primitive nucleus, then what I want to convey? Let me show you. So first point I shall write over here is that the cells who have primitive nucleus, not well-defined nucleus, right? And eukaryotic cells are those who have advanced nucleus. Now what I mean when I say primitive, I mean to say that when the genetic material which is represented by chromosomes are diffused in the cytoplasm, histone proteins are absent and that genetic material is not separated from the cytoplasm with the help of nuclear membrane. So absence of nuclear membrane. Second point, absence of histone proteins. And third point, genetic material chromosomes diffused in the cytoplasm. These 3 points are the characteristic feature of primitive nucleus. So what do you mean by primitive nucleus? Nucleus, and such nucleus is actually known as nucleoid or genophore. So prokaryotic cell, instead of nucleus, they have nucleoid or genophore, where genetic material is diffused in the cytoplasm. First point. Second point, lack membrane, lack nuclear membrane. Third point, do not have histone proteins. So such genetic material is known as nucleoid or genophore, and these are the characteristic feature of primitive nucleus. Now what do you mean by advanced nucleus then? Advanced nucleus, just opposite. First of all, genetic material is not directly diffused in the cytoplasm. Rather, it is separated from the cytoplasm with the help of nuclear membrane, double membrane, nuclear membrane, and genetic material represented by chromosomes have histone proteins. So histone proteins found, tick mark. Nuclear membrane lack, no, present, tick mark. And genetic material is not diffused. So such genetic material which is characterized by these points are considered as advanced nucleus. Here you will use the term nucleus, rather here you are going to use the term nucleoid or genophore. Correct? So what are the two types of cells on the basis of the nucleus? Prokaryotic and eukaryotic. This I have just given you some basic difference. The major difference we are going to learn as and when we will study about prokaryotic and eukaryotic cell independently and individually. So now we are going to start with prokaryotic cells. What are prokaryotic cell? See, the cells which have what? Which do not have a well-defined genetic material. The cells which have a primitive nucleoid or genophore. This point you know. Apart from this, prokaryotic cells are comparatively smaller in size as compared to eukaryotes, and as a result they divide even faster. So as compared to eukaryotic cell, these are smaller in size and divide comparatively faster. They multiply at a higher rate. Prokaryotic cells, some examples of prokaryotic cells are like bacteria, mycoplasma, cyanobacteria, then actinomycetes, etc. So prokaryotic cells, they are characterized by bacteria, Cyanobacteria, Mycoplasma. Mycoplasma are also known as PPLO, pleuropneumonia-like organisms. Right? Moving ahead. Now, prokaryotic cells, I have already told you, they may vary in shape, size and functions. So they greatly varies in terms of shape and sizes. There are 4 distinct shapes of prokaryotic cell, and on the basis of shapes, bacteria can be of 4 types. The 4 distinct shapes of a prokaryotic cell are bacillus, means rod-like, coccus means spherical, vibrio means comma shaped, and spirillum means spiral. All these points you have done in the kingdom Monera, so I'm not going to repeat it over here. You can just take a reference from lecture number 2 on the chapter biological classification, where I am teaching kingdom Monera. Correct? Yes or no? Will you take the reference from there? Okay, very good. Now, prokaryotic cells, my dear students, they have cytoplasm. Agreed. But their cytoplasm do not have compartmentalization. That means the cytoplasm of prokaryotic cell lacks membrane bound cell organelles. Correct? So next point which you should know with respect to prokaryotic cell is that cytoplasm lacks membrane bound organelles. However, non-membrane bound organelle like ribosome is found in the prokaryotic cell, and that ribosome is of 70S type. So however, they have 70S type of ribosome. Ribosome is basically, students, non-membrane bound cell organelle. Correct? Now, moving ahead. My dear students, you know that prokaryotic cells, they do not have a well-defined genetic material, but they have nucleoid, right? Now, apart from nucleoid, prokaryotic cells exclusively have some extra chromosomal self-replicating genetic material called plasmids. So plasmids are also found in the cytoplasm of a prokaryotic cell, which is absent in eukaryotic cell. Correct? What are plasmids? Plasmids are extra chromosomal self-replicating genetic material, right, which gives some extra phenotypic benefit to the bacteria or prokaryotic cell. So these are certain features which you have to remember, and for some more features you can see the lecture number 2, biological classification. Now, moving ahead. Now we are going to talk about the more structural organization of a prokaryotic cell. Now if I talk about the structural organization of prokaryotic cell, so first I'll be drawing over here all the structural features which I'm going to teach you, and then we are going to study all those features one by one. So first of all, I'm going to draw a rod-shaped bacteria, a rod-shaped prokaryotic cell, right? See everyone. Bacteria is characterized, or prokaryotic cells are characterized by 3 wall layers. The 3 wall layers from outer to inner are glycocalyx, cell wall and cell envelope. So here I have drawn one membrane, then I have to draw another membrane, another layer, and then I will draw the third layer. So this is the third layer which is also present around a prokaryotic cell. So overall you can say that prokaryotic cell is characterized by the presence of 3 wall layer. the outermost being glycocalyx. Inner to glycocalyx is cell wall, and cell wall encloses the innermost, that is cell membrane. Now, inside cell membrane will be what? You know, the membrane encloses the fluid material which is known as cytoplasm. So, it encloses the main arena of cellular activities called cytoplasm. Tick. Inside the cytoplasm is present diffused genetic material, not surrounded by nuclear membrane. And this is double-stranded, you can see, circular and single DNA, which is known as nucleoid or genophore. Apart from this, I have told you that bacteria, prokaryotic cell, is also characterized by the presence of some extra genetic material called plasmids. Right? Now, the cytoplasm do not have compartmentalization, they do not have membrane bound cell organelles, but they will have some non-membrane bound structures called ribosomes, and the nature of ribosome is 70 S types. Correct? Now, what is the role of ribosomes? My dear students, ribosomes are responsible for protein synthesis. However, for protein synthesis, these ribosomes need mRNA. Yes or no? So, number of ribosome, they get associated with mRNA present in the cytoplasm to form the structure called polyribosome that helps in translation. So, this is your mRNA, and this structure is polyribosome. What is polyribosome? mRNA along with many ribosomes for translation process. Will you remember this thing? Polyribosomes are also known as polysomes. Will you remember this point? Very good. Now, moving ahead. Now, this plasma membrane is a living layer. This plasma membrane shows some invaginations. It shows some infoldings either in the form of vesicles or in the form of tubes or in the form of lamella. These infoldings of plasma membrane are known as mesosomes. Function I will teach you when I will teach you plasma membrane in detail. Right now, I am just showing a picture of a prokaryotic cell. So that first picture is clear in mind. Then all structures, details of all structures you will be able to understand in a better way. So first I am telling you all the things which we are going to study in detail one by one. Right? Apart from giving rise to mesosomes, the cell membrane also have the capacity to give rise to locomotory organ called flagella. They can also give rise to some hair-like outgrowths that helps in the attachment of the bacteria called fimbriae. Right? One by one, we will study about all these things in detail. So this is how the structure of a typical bacteria looks like. Now, apart from all these things inside the cytoplasm, bacteria can have one more thing, and that additional things are known as inclusion bodies. Now, Cytoplasm do not have membrane-bound cell organelles. However, they can have some non-membrane-bound structures. Apart from non-membrane-bound structures, for storage purpose, these bacterial cells, these prokaryotic cells, they form some inclusion bodies in the manner of granules, like cyanophycean granule, phosphate granule, sulfate granule. So these granules are basically inclusion bodies that are responsible for the storage purpose. These are dead bodies. Clear? So this is how overall a bacterial cell looks like. Now let's discuss one by one all these structures which I have mentioned over here. First, starting from the cell envelope, that is the boundaries of the cell. How many cell envelope are there? Cell envelope is characterized by cell envelope consists of a tightly bound 3 layers. What are those 3 layers from outer to inner? Glycocalyx, cell wall and cell membrane. Can you see this in the diagram? All the 3 layers very clearly? Right. So now we are going to study all these 3 layers of the cell envelope one by one, outer to inner. Now, all these 3 layers of the cell envelope, they have some specific function. Apart from those functions, the overall function of this whole cell envelope is to act as a protective unit. All these 3 layers together, they help in protecting the bacteria from any kind of injury. Right? Almost each layer of the envelope has its own distinct function, but overall they act as a single protective unit. Clear? Very important line. NCERT states this line, NEET will ask question. Right? So will you not make any problem in this? And will you pay attention to all the things which have been told to you? Yes, very good. So behave like a very good students, studious ones, and the ones who listen to their teacher, be it an online teacher or offline teacher. Both are teachers, my dear student. And if you're paying attention to what your teacher, what your elder is saying, then surely you are you're taking your life in a positive way. As I started my class today with a simple thought. And what was that thought? And that thought was that whenever you get up in the morning, get up with some positive thinking, with some positive thoughts, and then you see the change in your whole day. You will remain positive whole day. Positive things will come around you. Even in negative aspect, you will find some positive aspects, right? But if you get up in the morning with some hard thinking and like, oh, again a new day started, again study, school, college, tuitions, online teaching, etc., then your whole day will be wasted. You'll always feel lethargic and low in energy. So try to make a change in your own life with one simple day. Try this simple thing. Get up in the morning with some positive thought and then check difference in your life. Okay? So coming back to the topic, first outermost layer we are going to talk about is glycocalyx. So first point in glycocalyx, you have to remember that it's the outermost layer, right? Now this outermost layer, my dear students, can be present in 2 forms. That is, either this layer can be present in the form of smooth, slimy, mucilaginous layer that is called as slime layer, or it may be present in the form of thick, tough, resistant layer called capsule. So your glycocalyx can be present in 2 forms, either in a mucilaginous sheath-like covering called slime layer, or it can also be present in the form of thick, tough, resistant capsule. Clear? Now, if glycocalyx is present in the form of slime layer like in cyanobacteria, cyanobacteria is one of the example of prokaryotic cell whose glycocalyx is present in the form of slime layer. So the slime layer has function protection is there, but apart from protection, it performs one distinct property. It has one distinct function, that is to protect the bacteria from dehydration. So basic specific function of slime layer apart from protection is to protect the bacteria from dehydration. Clear? Talking about capsule. Capsule is also a glycocalyx. However, when glycocalyx is present in the form of a thick, tough, resistant layer, and if capsule is present in the bacteria, then apart from protection, it gives bacteria resistance towards the host immune system. So apart from protection, glycocalyx as capsule immunes the bacteria from the immune system of the host. So capsule function is protect the bacteria from host immune system. Correct? Yes or no? Okay, very good. So what is glycocalyx? Outermost layer may be present as slime or tough capsule. General function is protection, and specific function also I have told you. Now, moving ahead towards the next layer, which is known as cell wall. Inner to glycocalyx is cell wall. The cell wall is a dead layer. It's a fully permeable layer. Fully permeable means it will let all the material in and all the material move out. So it is a dead layer, inner to glycocalyx, fully permeable. And what this layer is made up of? It is made up of peptidoglycan, which is commonly known as murin. So if I talk about peptidoglycan, then what is this peptidoglycan? Peptidoglycan is the basic unit of the cell wall of bacteria, where peptidoglycan is made up of proteins and sugar. Peptide term means protein and glycan term means sugar. So the two different sugar units that join together to form peptidoglycan is NAG and NAM. NAG stands for N-acetyl glucosamine, whereas NAM stands for N-acetyl muramic acid. If I talk about protein, then there are 4 amino acids which joins together to form peptidoglycan, that is L-alanine, D-alanine, L-lysine and D-glutamic acid. So these are the 4 amino acid components and 2 sugar units that join together to form one peptidoglycan, and number of such units they cross-link to form the cell wall. So the cell wall of bacteria is made up of murin or peptidoglycan. Correct? Now talking about the function of cell wall. Cell wall provides protection, no doubt. Apart from protection, the main function of cell wall is to protect the cell from bursting. It maintains the osmolarity within the cell. It does not let excess of water enter inside the cell. So it protects the cell from bursting. Right? This is the it is flexible. Not flexible basically. It is going to maintain the shape of the cell. It is going to maintain the water balance, you can say, and it will avoid the bursting of the cell. Correct? So what is the main function? It protects the cell from bursting and also maintains the shape of the cell. Apart from this, cell wall also protects the cell from desiccation or injury. Right? Very good. Overall, all prokaryotic cells, they have cell wall except Mycoplasma. The smallest bacteria lacks cell wall. PPLO lacks cell wall. So this is the next point which you should remember is that it is found around all bacterial cells except Mycoplasma. Right? Now, cell wall is sensitive to antibiotic penicillin. That means if a bacteria who is having cell wall and if you inject that bacteria with penicillin, that this penicillin antibiotic is going to destroy cell wall, thus killing the bacteria. Now, Mycoplasma lacks cell wall. So can we say that Mycoplasma will be resistant to the attack of penicillin? Yes, Mycoplasma are resistant to penicillin. Done? Why? Because they lack cell wall. Moving ahead, it also provides one more function I have added, that cell wall also provides rigidity to the cell. Correct? Now, moving ahead, this much you have to do for cell wall. Now, moving ahead towards the third layer of the cell envelope, that is cell membrane. But before talking about cell membrane, there was a scientist called Christian Graham. There was a scientist called Christian Graham who performed staining experiment and divided bacteria into two categories. He divided prokaryotic cell into two categories as gram-positive bacteria and gram-negative bacteria. How and why? See, when Christian Graham was performing his staining experiment, he saw two categories of bacteria. What were those two categories of bacteria? One group of bacteria was having three wall layered, as I have told you, glycocalyx, cell wall and cell membrane. But some group of bacteria were such who were having four wall layered. There was one additional wall layer in certain group of bacteria, and those four wall layered are glycocalyx, outer membrane, then cell wall and then cell envelope. Right? So you can say that with the help of staining experiment, Christian Graham divided bacteria into two broad groups as gram-positive and gram-negative. Why? What he saw? He saw two groups of bacteria, gram-positive and gram-negative. Gram-positive are those bacteria which have normally three wall layered, that is cell membrane, which is lipid bilayer, you all know. Correct? Followed by cell wall and followed by glycocalyx. So let me move it from here. Let me write it over here. Correct? So he found three wall layered normally in gram-positive bacteria, that is plasma membrane, cell wall and glycocalyx. No issues. But in some group of bacteria called gram-negative, he found four layered. Here it is three wall layered envelope. But in case of gram-negative, he found four wall layer, and those four wall layers are the innermost plasma membrane, followed by cell wall. Now in between cell wall and glycocalyx there was an additional layer called outer membrane. So this is glycocalyx, this is cell wall, this is cell membrane, but there's one additional layer called outer membrane between glycocalyx and cell wall. Correct? Because of this reason, gram-negative bacteria were not ready to accept the stain at the very first time. So Christian Gram, on the basis of their cell envelope, divided prokaryotic cell into two broad categories. Have you understood that what is the basic difference between gram positive and gram negative? So moving ahead, let's read what is written on this page. Bacteria can be classified into two groups on the basis of their difference in their cell envelope, correct? And the manner in which they respond to the staining procedure performed by Christian Gram. Christian Gram, on the basis of a staining experiment, divided bacteria into positive and negative. Now I'm going to show some differences between gram positive and gram negative which are important for you and you can remember. Number one, already told you, what are gram positive bacteria? Those bacteria which have three wall layered. That means glycocalyx followed by cell wall and followed by cell membrane. But gram negative bacteria, they have four wall layered cell envelope, which is glycocalyx followed by outer membrane, then followed by cell wall, and then followed by the innermost cell membrane. Right? Now, due to the absence of one additional layer in gram positive bacteria, they have thick cell wall. So gram positive bacteria are characterized by the presence of thick cell wall. On the other hand, gram negative bacteria, they have comparatively thin cell wall. Now, since in gram positive bacteria you find thick cell wall, so that means amount of peptidoglycan will also be high. So in case of gram negative bacteria, amount of peptidoglycan is high. But obviously in case of gram negative bacteria, that amount of peptidoglycan is going to shrink, it is going to become less. So they have less amount of peptidoglycan. Yes or no? Outer membrane is present in gram negative bacteria which is made up of lipopolysaccharide. So outer membrane, due to the presence of outer membrane, gram negative also have high content of LPS, lipopolysaccharide. So outer membrane is made up of lipopolysaccharide, that is lipid and sugar. Whereas if I talk about gram negative bacteria, then such layer is absent in them. So these are certain differences for gram positive and gram negative which you should remember, and they are going to help you in your question solving. Now with this, cell wall part is over. The functions, what it is made up of, types of bacteria on the basis of cell envelope, all these things are over. Coming to the third layer from outer to inner is cell membrane. Now we are going to study about what is cell membrane. Are you all ready to study about cell membrane? Yes, ma'am. Very good. Chalo. So let's talk about cell membrane, which is also called as plasma membrane. It is the innermost layer. First point. Apart from innermost, Unlike that of cell wall, it is living layer. Unlike that of cell wall, it is selectively permeable. Selectively permeable means it is going to allow some selected molecules only to move in and out of the cell. Unlike that of cell wall, which was fully permeable, letting all the material in and letting all the material out. So cell membrane is not going to function in that manner. It is selectively permeable, so it is only going to allow some material, some selected molecules to move in and out of the cell. Clear? First 3 points are clear to everybody. Fourth point. Now, what the structure looks like. So if I talk about the structure of plasma membrane, it is represented by lipid bilayer embedded with protein. Same model, fluid mosaic model. Remember students, चाहे eukaryotic cell हो or be it prokaryotic cell, there is no similarity in between these two cells except their structure and function of plasma membrane. Structurally, plasma membrane of both pro and eukaryotic cell is same. That is, it is represented by lipid bilayer along with proteins. The model which was explained by Singer and Nicholson, fluid mosaic model. I will talk about this model later on when we will start about eukaryotic cell. But here you should just remember one point for time being, that the plasma membrane is structurally similar to a eukaryotic cell plasma membrane. That is, it is made up of lipid bilayer along with protein. Correct? The basic function of plasma membrane, if I talk, then the basic function of plasma membrane to allow bulk transportation of material. It allows bulk transportation of material in and out of the cell. But that bulk transportation is also going to be selective in nature. Correct? Because this layer is selectively permeable. Right? Now, since I told you that plasma membrane is living, so it can give rise to certain structures, and the most famous structures which are given by plasma membrane or which arise from plasma membrane are pili, fimbriae, flagella. These three are the outer extension, and one inner extension is mesosomes. Right? So how many extensions are there? 4. 3 outer and 1 inner. One by one, little bit, not much detail you have to do for all these 4 structures. So first coming to fimbriae. What are fimbriae? Fimbriae are hair-like outgrowths which occur in tuft, and they help in attachment of the bacteria with the substratum. They are made up of protein fimbrine. So fimbriae, first of all, they are made up of fimbrine protein, first point. Second, they are hair-like outgrowths. Correct? क्या करेंगे? They help in the attachment of the bacteria to the substratum. Helps in the attachment of bacteria to substratum. Clear? Now, talking about second extension. About this also I have talked in the chapter biological classification. Pili. Pili are also hair-like outgrowths made up of protein pillin, and their role is to help in sexual recombination through conjugation method. Pili, they help in exchange of genetic material between the two bacteria. These pili, they form a conjugation tube which let the transmission of genetic material from one bacteria to another. This method is called as conjugation. So what pili helps? They help in conjugation. Correct? Moving ahead, talking about flagella. But before flagella, I have mentioned mesosomes. So what are mesosomes? Mesosomes are the infoldings. They are the infoldings which can be either present in the form of vesicles or in the form of tubes or in the form of lamella. So mesosomes are the infoldings present either in the form of vesicles or in the form of tubes or in the form of lamella. These mesosomes, my dear students, which are the infoldings of plasma membrane in prokaryotic cell, are of great importance. Why? Because these mesosomes, they contain respiratory enzymes. Thus, they are analogous to eukaryotic mitochondria. What mitochondria in a eukaryotic cell performs, that is respiration, same function for prokaryotic cell mesosome performs. You know, prokaryotic cell, they do not have mitochondria. So who is going to perform their respiration? Mesosomes. Apart from respiration, these mesosomes also help in DNA replication. They help in cell wall formation. Correct? So what are the roles of mesosome? Number one, help in respiration. Number two, they help in fission. Third, they help in DNA replication. Correct? So these are the functions, very important functions performed by the mesosomes, and they contain respiratory enzymes. So will you remember all these points related to the extensions of plasma membrane? Now one is still left, that is known as flagella. So flagella is a locomotory organ. The number of flagella may vary from 1 to many. They are made up of flagellin protein. So what is flagella? Flagella, first of all, it is an extension of plasma membrane. You know that. Apart from this, they are made up of flagellin protein. Number one point. Second, they help in motility. If flagella is found in a prokaryotic cell, that prokaryotic cell will show movement. If not, then it will not be motile. Structurally, if I talk about, then flagella is structurally represented by three parts. How many parts? Three parts. That is lowermost basal body, then hook, and the part which you actually see, wavy part, is known as filament. This basal body is made up of rods and usually 4 rings. That is L, P, S, M. Tick. Little bit I'll show you the diagram, but even if you ignore this diagram, then it's not important from the point of NCERT. NCERT has not given also, and in NEET also they don't ask question related to this. But still I am showing you it. Suppose this is plasma membrane and suppose this is cell wall. So the flagella is going to start from it is going to originate from the plasma membrane itself. So first part is basal body, which is made up of what? Rod And around this rod there are how many rings? There are 4 rings. Now from the rod then arises hook, and then this hook give rise to the filament. So hook is a hollow structure that connects basal body to the filament. Filament is that structure which you see moving, and the movement is controlled by the basal body, which is represented by rod and 4 rings. So this is your basal body, this part is hook, and this part is filament. Even if you don't do this part, no issues. Just remember what I have told you, made up of flagellin protein, motility, and this much. Will you do this much? So this is about your plasma membrane. Structure of plasma membrane I will discuss when I will start teaching eukaryotic cell. Clear? Apart from this, in a prokaryotic cell, you also see ribosomes, which are non-membrane bound cell organelle, and the nature is 70S. Then you also see inclusion bodies, which are the dead non-membrane bound bodies responsible for storage of material like phosphate granule, sulfur granule, cyanophycean granule, gas vacuole. In some cyanobacteria and purple sulfur green bacteria, gas vacuoles are found. Gas vacuoles are not true vacuoles, they are pseudo vacuoles. They are non-membrane bound inclusion body responsible for storing gas and providing buoyancy. So these are the things which you have to remember for a prokaryotic cell. Rest you can read it from NCERT. If you go through this lecture, then I can 100% guarantee you're not going to miss any single point from your NCERT. In fact, apart from NCERT, I have given lot of information which is extra, and it is equivalently important from the NEET point of view. So if you go through this lecture, it is really going to be very beneficial and very helpful. Then you can try solving the questions. If you face any trouble in solving the questions, you can anytime post those questions in the comment section, and 100% I can guarantee I'm going to answer all your questions and queries. So please stay in touch, stay connected, and keep learning, and don't waste time. It's a high time to study. Now, one question is there on your screen. I hope all of you can see. Which of the following helps in sexual recombination? So options are fimbriae, flagella, pili, and mesosomes. Right now only you did this question. Fimbriae, attachment, no. Flagella, locomotion, no. Mesosome, respiration, no. What is left? Pili. Pili, they help in sexual recombination by conjugation method. So answer of this becomes B. And with this, your prokaryotic cells are over. Now we are going to start with eukaryotic cell. Now I hope that this question is clear to everybody. With this, prokaryotic cell is over. Now moving ahead with the next group of cells, that is eukaryotic cell. Eukaryotic cell, students, means those cells which have a well-defined genetic material. That means the genetic material represented by chromosomes, they are separated from the cytoplasm with the help of a membrane called nuclear membrane. Second, the packaging of the histone, the packaging of the chromosome, the genetic material is done with the help of histone proteins. So the eukaryotic cells are those which are having, indirectly we can say, they are possessing true nucleus. So eukaryotic organisms or eukaryotic cells are those which possess true nucleus, well-defined genetic material. Under eukaryotic cells, organisms except kingdom Monera, Protistians, fungi, Animalia, Plantae, all these are made up of cells which are eukaryotic in nature. So eukaryote includes all the members of different kingdom except kingdom Monera. because kingdom Monera have only bacteria, and bacteria are represented by prokaryotic cell. Correct? Moving ahead with the second point. Now, in eukaryotic cell, apart from the main genetic material, their cytoplasm, which is the main arena for all cellular activity, is also divided into different chambers called membrane bound organelles. So eukaryotic cell, they show extensive compartmentalization due to the presence of membrane bound organelles. They may be single membrane or they may be double membrane. This structure was absent in case of prokaryotic cells. Prokaryotic cells, if you remember, they only have non-membrane bound organelles like ribosome. But eukaryotic cells, they have membrane bound organelles present in them. Third point, I already told you, they possess a well-defined genetic material, the genetic material which is enclosed within the nuclear membrane. Now, moving ahead, if I talk about eukaryotic cells, then eukaryotic cells, my dear student, they have exclusive presence of tubulin protein, right? This tubulin protein is found in the cytoplasm of eukaryotic cell that helps in making certain locomotory structures like cilia and flagella. Apart from this tubulin protein also help in the manufacturing of cell organelle centrosome. So tubulin protein is very important, that is found in the eukaryotic cell, and it is responsible for maintaining the cytoskeletal structure, which is absent in case of prokaryotes. So presence of cytoskeleton structures like microtubule is actually due to the presence of tubulin, and this protein helps in maintaining the motility, cell division, cell growth, cell shape, etc. Correct? Now, the cytoplasm of eukaryotic cell also shows movement called cyclosis. Unlike that of prokaryotic cell, the eukaryotic cell cytoplasm can show circular movement, which is known as cyclosis. Right? Now, eukaryotic cell, they also have ribosome, but their cytoplasmic ribosome is ATS type. So you can mention in the bracket that the cytoplasmic ribosome in case of eukaryotic cell is ATS. However, some cell organelles which have prokaryotic in origin like mitochondria and chloroplast, they also have 70S ribosome. But the main ribosome of eukaryotic cell is ATS type. So these are certain general features, including one more, plasmids, the extra genetic material which you observed in case of prokaryotic cell is absent in case of eukaryotes. We do not have this extra chromosomal genetic material, that is plasmid. Presence of plasmid is a characteristic feature of prokaryotic cell. Clear? I hope all the points which are written on the board are clear to each and every one. If you remember these points, that means you can easily differentiate between prokaryote and eukaryotic cell. Now, in this chapter, we are going to study two eukaryotic cell, plant and animal. So first, we are going to study the difference between eukaryotic plant cell and eukaryotic animal cell, and then we will move ahead with the structural organization. So first, few basic difference which we all know, I am going to repeat them again, so please pay attention. If I talk about plant cells, then what is the major character or feature that differentiate from between plant and animal cell? That feature is the presence of cell wall. Plant cells, they are characterized by the presence of cell wall, but animal cells may, this cell wall is completely absent. So we can write that plant cell, cell wall is uniquely found in them. However, animal cells, they lack cell wall. First point. Moving ahead to the second point. Now, this is about the envelope. So, in case of plant cell, the outermost layer will be cell wall. But in case of animal cell, the outermost layer will be cell membrane. Now, coming inside the cell, cytoplasm is there. However, in case of plant cell, the center of the cytoplasm, the 90% of the space of the cytoplasm is occupied by vacuoles. So, vacuoles occupy 90% of the total cellular space. As a result, the cytoplasm as well as the nucleus, in case of plant cell, they shift towards the periphery, obviously. Now, it's something like this. Suppose this is a plant cell with a cell wall as the outermost layer, followed by cell membrane. Now, inside is whole cytoplasm with a well-defined genetic material. But the 90% of the space of the cell will be occupied by vacuole, because of which nucleus and cytoplasm will shift towards the periphery. Yes or no? Correct. So, here third point you can write that cytoplasm and nucleus shifts towards the periphery. Any confusion to anybody? No. Very good. So now moving ahead. Coming to animal cell. Animal cell, first of all, vacuoles are either very small or absent. Hence, the central space of the cell is occupied by a well-defined nucleus and densely cytoplasm are present. Clear? Moving ahead. Now, coming to the compartments present in the cytoplasm. So, cytoplasm, they have one special cell organelle called plastids, which are absent in case of animal cell. Rather, animal cells, they have centrosome for cell division, which is absent in case of plant cell. So, two difference you can mention. Number one is that what? They lack centrosome, but they have plastids. Correct? So, they have plastids. They do not have plastids, but they have centrosome. So, now, I hope all these points are clear to everybody. One more you can mention that in case of plant cells, Golgi complexes are represented by number of cisternas. However, they are not interconnected. Thus, in case of plant cells, Golgi bodies are given some different name like dictyosomes. But if I talk about animal cells, then the Golgi body is called as Golgi body only, as the number of cisternas, they are interconnected to each other. So this point also you can mention as a difference between plant and animal cell. Now moving ahead, and now let's talk about the envelope of eukaryotic cell. Both plant and animal, we are going to take them together. Along with the unique features, we are also going to study their common features. So first, starting with their envelope. So here I'm going to talk about that first envelope, first layer of the cell envelope, which is common in both prokaryote as well as eukaryote, and that is, prokaryote as well as eukaryote, absolutely correct. And in eukaryote also, it is found in all the cells, that is cell membrane. Now, if you remember students, I have already told you when I was teaching you prokaryotic cell that in between prokaryotic cell and eukaryotic cell, there is hardly any similarity except the structural organization of plasma membrane. In prokaryotic cell as well as in eukaryotic cell, the plasma membrane is represented by fluid mosaic model proposed by Singer and Nicholson in the year 1972. This fluid mosaic model tells that plasma membrane is present in the form of lipid bilayer embedded along with proteins. Now, you must be thinking, ma'am, that since 1972 only are we aware about plasma membrane? What about before that? What about in 1970s and before that time? So, my dear students, there are many scientists who tried to propose some basic structures of plasma membrane. The many theories came even before fluid mosaic model, but soon all those theories were discarded when Singer and Nicholson proposed their theory. So you have to only remember one theory, which is the most accepted theory of fluid mosaic model. It was discovered by Singer and Nicholson while they were working on human RBC. After the discovery of ultra electron microscope, these two people, by using human RBC cell as a specimen, they discovered, they explained the entire structure and function of plasma membrane. So are you all set to see that how the structure of plasma membrane actually looks like? So chalo, let's get started. Now, see, first, the detailed structure of the membrane was studied only after the advent of electron microscope. And before this, many theories were proposed, but they had some or the other problems and disadvantages in their theory, so they got rejected. The most accepted model which we are going to study is fluid mosaic model, which was proposed in the year 1972 by the scientist Singer and Nicholson. Right? According to this model, the plasma membrane is represented by lipid bilayer embedded along with proteins, and some amount of carbohydrate is also found. Right? Some percentage of carbohydrate is also there on the plasma membrane, present either in the form of glycolipid or glycoprotein. But the two major components that are making the plasma membrane are lipid and protein. They were studying on human RBC, and they found that in human RBC, the amount of lipid was approximately 40%, whereas the amount of protein was 52%, and remaining was carbohydrate. Clear? Now let's see the structure of this human RBC or the structure of plasma membrane in detail. So clear to everybody? Now we are going to see the structure of plasma membrane in detail. Right? So okay, now let's see what the structure of plasma membrane is actually all about. So first for understanding the structure of plasma membrane, I am going to first draw a cell, right? Suppose this is a eukaryotic cell. Pay attention everyone, okay? Suppose it's a eukaryotic animal cell which I am trying to draw, which is having only one outer membrane as plasma membrane. Inside it encloses cytoplasm and a well-defined genetic material along with other compartments in the form of membrane-bound organelles. Now, outside, if I talk about the plasma membrane, is aqueous environment, intercellular space, And if inside plasma membrane I talk about is cytoplasm, which is also richly made up of water. Hence inside intracellular region is also aqueous in nature. This is intracellular region and this is extracellular region. Yes or no? Correct. Now actually you have to study this structure in detail. So according to fluid mosaic model, the plasma membrane is double layered. It is represented by two layers which are made up of phospholipid, lipid associated with phosphate. So it is, this plasma membrane is represented by two layers which are actually made up of lipid, right? And which type of lipid? Phospholipid. Right? This is how the lipid bilayer is arranged in case of plasma membrane. Now, this region is intracellular, right? And this region is extracellular. So both intra and extracellular region are aqueous in nature. Remember this thing, right? Now you can see that lipid bilayer, how it is represented. Now let me take out one molecule of this lipid layer outside. So each molecule of lipid in the bilayer is represented by two components. One which you can see in spherical manner and another which you can see in like leg-like manner, right? So the spherical part is known as head, whereas the lower leg part is known as tail. Now you can also see one thing, that the head is facing both the aqueous region, extracellular as well as intracellular, whereas tail is well protected by the head from the aqueous environment. This means surely head is hydrophilic in nature, it is polar in nature, whereas tail is non-polar or hydrophobic. It is made up of hydrophobic hydrocarbons. So you can write one thing, that head is polar or hydrophilic, whereas tail is non-polar or hydrophobic. That's why tail is well protected by the head from the two aqueous environment, that is both extracellular and intracellular. So this thing you have to remember, that what is head representing and what is tail representing. Now apart from this lipid bilayer, on this lipid bilayer rather, proteins are embedded. Fluid mosaic model. In the name fluid mosaic model, fluid represents, where it is, this fluid represents for lipid and mosaic represents for protein. Singer-Nicholson well said that plasma membrane is represented by a sea of lipid in which proteins are present as icebergs. So mosaic is actually protein and fluid is actually lipid. So this is the fluid on which your proteins are present in the form of icebergs in a sea. Now, if I talk about proteins, then there can be two types of proteins. Some which are directly attached on the lipid bilayer, that means which are just present on the surface. They will be very easy to extract. Such proteins are called as extrinsic protein. Extrinsic means external proteins which are just attached on the membrane, just present on its external surface. These proteins are easy to extract. On the other hand, some proteins you will find embedded inside the membrane. Those set of protein will be called as intrinsic protein. The proteins which are embedded within the membrane. Such proteins which are embedded will not be easy to extract. Correct? You can very well understand this. Now, intrinsic protein again can be of two types, some partially embedded or some totally embedded. Now, I am going to show you with the help of the diagram. So, this is the outer layer. This is also the outer region of the inner layer. So, this is A and this is B. Both A and B are extrinsic protein. Extrinsic does not mean that the protein which are present on the lipid bilayer facing the extracellular region. No. Lipid bilayer है, उस पर लगे हुए कोई भी external protein may be on the upper lipid bilayer or on the lower lipid bilayer. So A and B both are extrinsic protein. Now, if I talk about intrinsic protein, then they can be of two types. Either partially embedded or totally embedded. Partially means like this. So can you see this pink color protein? It is going inside the membrane but not completely. So it is partially embedded but still it is intrinsic. And some are totally embedded. The proteins which you can see through the lipid bilayer from the upper end as well as from the lower end. Then these both C and D, they both are intrinsic protein. They both are difficult to remove, but some are partially embedded and some are totally embedded. However, they both are intrinsic. Clear? Now, intrinsic protein में the one which is completely embedded, totally embedded, again can be of two types. So, totally embedded protein again can be of two types, either as tunnel protein, also called as porins, or channel or carrier protein. Tunnel proteins are those intrinsic proteins, those completely embedded intrinsic protein which are acting like a opening. They are just like a pore. Like for example, if I want to draw, now just see, suppose this is plasma membrane, right? On this some extrinsic proteins are present, some intrinsic proteins are present. Now intrinsic protein can be of two types, some totally embedded or some partially embedded. Now totally embedded, one type of totally embedded is intrinsic. One type of totally embedded is tunnel or porin or pore. So that means the protein which is hollow from inside like this. So this is the tunnel protein. The protein which is hollow from inside, it is acting like an opening in the plasma membrane so that hydrophilic substances can easily move here and there across the membrane. Correct? So what are tunnel proteins or porins? These are those openings on the plasma membrane which are actually letting the movement, the easy movement of hydrophilic substances. Clear to everybody? Okay. On the other hand are carrier proteins. They are also intrinsic, but they are solid proteins. So this is actually your tunnel protein and this is your carrier protein. Carrier protein are solid. That means any material cannot directly pass through this protein. So these carrier proteins have specific receptors present on their surface to which molecule bind. Then they move in 180-degree manner. Then they leave those molecules on the other region and again come back to their original position. But tunnel proteins, they do not show this movement. They are simply openings. They're like a gate. There's a room and in the room there is one complete gate. So that gate, if it is open, anybody can pass through that opening. Right? Yes or no? So it is just the gate of a room is equivalent to tunnel protein on the plasma membrane. It's just like a pathway, it's just like an opening. Any hydrophilic substances can move through them. But carrier protein, they are not like openings. They are simply having receptors on their surface to which molecules bind. Then they move either inside or outside the cell. Clear? So what are proteins? Proteins are those chemicals which are present on the plasma membrane. They can be two types, extrinsic, intrinsic. Intrinsic can be either partially or totally embedded. If they're totally embedded, again they can be of two types, carrier protein or tunnel protein. Overall, the function of this protein, the function of the membrane is basically allowing the selective movement of molecules inside and outside the cell. So the basic function of plasma membrane is bulk transportation. Transportation of molecules either inside or outside the cell with the help of proteins if the molecules are hydrophilic or charged particles, and directly also by the method of simple diffusion if the molecules are hydrophobic. Clear to everybody that how the structure of plasma membrane actually is? So it's fluid mosaic model in which fluid represents lipid and mosaic represents protein. Year 1972, proposed by Singer and Nicholson, right? I don't know what it got rubbed. Yeah, they were studying on human RBC. The chemical composition of human RBC and the types of proteins. So I hope the entire structure of plasma membrane is crystal clear. Now, moving ahead towards the next layer of cell envelope, which is uniquely found in either plant cells, some protisteans like euglenoids and fungi, but they are absent in case of animal cells. So this layer is known as cell wall. Now I'm going to teach you something about cell wall. Let's start. So what is cell wall? First of all, it's a non-living rigid layer. Unlike that of plasma membrane, students, plasma membrane was a living layer. It was selectively permeable, right? Non-rigid. But if I talk about cell wall, it is non-living, rigid layer, and it is fully permeable, will allow every material to move in and move out. Correct? Yes. Now, so first point, I hope it's clear. Right? Second point, cell wall, the main function of cell wall is to maintain the shape as well as protect the cell from bursting. They maintain the water balance inside the cell. They prevent the bursting of the cell. Plus, they also maintain the shape of the cell, provides rigidity and strength as well. Correct? They also protect the cell from the undesirable macromolecules. They don't let pathogen enter inside the cell. So they act as a barrier to undesirable molecules. I hope the function and where these cell wall are found is clear to everybody. Now, you know that cell wall is found in protisteans like in some protisteans like your diatoms. Euglena do not have cell wall. They lack cell wall. Some protisteans, I meant to say like diatoms, who have siliceous cell wall. Dinoflagellates, they have cellulosic cell wall. But Euglena do not have cell wall. Right? Okay. Now, moving ahead on the basis of composition. In different groups of organisms, cell wall is made up of some different chemicals. Like if I talk about monerans, then their cell wall is made up of peptidoglycan. Coming to eukaryotic part, there are 3 organisms who have cell wall and their composition is also variable. That is fungi, plants, and in plants, algae. If I talk about first plants, then in plants, it is richly made up of cellulose. It's a polysaccharide, hemicellulose, and pectin sugar. If I talk about algal cell wall, it is also richly made up of cellulose, but apart from cellulose, they also have some sugar like galactans and mannans. Right? And there are some algae whose cell wall also have deposition of calcium carbonate. So algal cell wall, usually it is made up of cellulose. Apart from cellulose, it also richly contains galactans and mannans. And in some algal cell wall, minerals like calcium carbonate are also reported. Right? Plant cell wall, everybody knows cellulose, hemicellulose and pectin. Third organism to possess cell wall in eukaryotic cell is fungi. So fungal cell wall is chitinous. Chitin is actually what? It is a polymer of sugar NAG, N-acetyl glucosamine. It's a homopolymer of NAG. Number of NAG units, they join together to form chitin, and chitin makes the cell wall of fungi. Clear? So this is types of cell wall on the basis of composition. Now, moving ahead, types of cell wall on the basis of origin. If I talk about types of cell wall on the basis of origin in a cell, then there are two types, primary cell wall as well as secondary cell wall. What is primary cell wall? What is secondary cell wall? I'll tell you, but with the help of a diagram, and then we are going to write their points. Chalo, let's see. Now, there's a parent cell, correct, which has undergone division to produce two new daughter cells. Now, these two new daughter plant cells, they are initially young, they are immature. So, such initially young and immature plant cells, they have cell wall which is thin, flexible, living, cellulosic. This type of cell wall which is thin, young, flexible, living, this type of cell wall is known as primary cell wall. But with time, these daughter plant cells will also get mature, and with maturity, the primary cell wall is replaced by number of thick-walled, rigid, dead, non-proteinaceous, non-flexible, non-elastic secondary cell wall. So basically, when the cell is young, see, suppose this is a plant cell which I am drawing, a young plant cell. Initially, this is a young plant cell, right? So young plant cell is also having a cell wall, but this is primary cell wall, which is characterized by being thin, flexible, living, and immature. Flexible means it can grow, it is elastic in nature. Why? Because it has the presence of proteins like elastin and expansin. Correct? So this is your primary cell wall. Inside this primary cell wall is what? Plasma membrane. Plasma membrane encloses a huge vacuole because of which nucleus as well as cytoplasm gets towards the periphery. So this is actually a young plant cell. Now, when this plant cell will become mature, will try to become older, what happens? The extensin and expansin protein of primary cell wall allows primary cell wall to expand. Now, when primary cell wall is going to expand, what will happen? The space between cell membrane and primary cell wall will be created, and in this space will be laid down new cell walls called secondary cell wall, which will be non-flexible and non-elastic. Slowly and steadily, due to excessive expansion in the primary cell wall, the primary cell wall will get diminished, it will get vanished, and once the plant cell become mature, it will only have multi-layered secondary cell wall which will be thick, dead, non-rigid, as you have studied in the features of cell wall. Correct? So now what will happen? This primary cell wall will expand, It is going to increase the space between the cell wall and cell membrane. So now what will happen? Primary cell is moving out, right? Creating more space between the primary cell wall and plasma membrane. And now in this space, new secondary cell wall, multi-layered, 2 to 4 layered secondary cell wall will be laid. This is intrinsic growth, yes or no? Intrinsic growth. So this is your primary cell wall. This is secondary cell wall, but S1. Now, one more will be formed. One more layer of secondary cell wall that you can label as S2. Again, one more can be laid down, which you can label as S3. So these are basically secondary cell wall which are thick. Obviously, so many layers are there, so it will be automatically thick. Then rigid, non-living, because they do not have any proteins on them, so they will be non-living. Can they expand? If they are non-living, can they expand? No. So they are non-flexible. And they are showing intrinsic growth. Correct? A time will come that this primary cell wall will get vanished. Okay, I'm not going to vanish them, otherwise my whole diagram will get vanished. Okay, so a time will come when this primary cell wall will get vanished, and once the cell becomes completely mature, it will be characterized by a big vacuole and multi-secondary cell walls, whose main function will be to provide strength and rigidity to the cell. I hope on the basis of origin, the two types of cell wall, primary and secondary, is clear to everybody. Right? Okay, so now moving ahead. So this is about types of cell wall, first on the basis of composition and second on the basis of origin. Now you have understood the cell wall structure. But if I talk about number of cells, plants, fungi, they are multi-celled organisms. When multi-celled organisms are there, that means their body is made up of so many cells. Who is going to keep those cells together? Who's going to keep those cells attached? Like for example, there is a wall. You know, wall is made up of multiple bricks. Now, if you want to make a wall and then if you only keep the bricks without joining them, will that wall be of any use? You just kick that wall and that wall is going to break at once. Little bit wind will blow off that wall. Why? Because you have kept the bricks but you have not joined them. So a multicellular organism in plants is made up of number of cells, but those cells, they're just piling one above the other. If they're not joined, then even a single small wind can blow away that body and no multicellular organism will be of any use. Hence, each cell in plants, they are joined together with the help of calcium magnesium pectate called middle lamella. So now next topic is to understand middle lamella. So middle lamella is that cement. In order to make a wall, you need bricks as well as cement. Correct? So to make a full-fledged multicelled organism, you need cell as well as middle lamella. So what is middle lamella? It is a cementing substance that glues the two cells, that holds the two cells together. Correct? It holds the two cells together and it is made up of calcium and magnesium pectate. The formation of middle lamella, it actually happens during the time of cytokinesis only. When the cytoplasm of plant cell is dividing, It helps in the formation of cell plate. And that cell plate, after division, itself start acting as middle lamella. So you can write this point as well. Cell plate during cytokinesis, produced during cytokinesis, becomes middle lamella in future. Right? Now you can see the diagram. I'll show you. Suppose these two are mature plant cell. Mature plant cell, that means they will be only having what? Secondary cell wall and a large vacuole. So this is my one plant cell, this is my another plant cell. Right? Now, in order to keep these two cells together, we need the deposition of calcium magnesium pectate, that is known as middle lamella. So each cell from all the four sides, they are joined. Now here also one cell will be kept. Yes or no? So here there will be some middle lamella. Similarly here there will be one cell, again middle lamella. So in order to join the two cells together, there's a deposition of middle lamella, which is calcium magnesium pectate. Now imagine there's a room with 4 doors, right? No, imagine that there's a room without any door, without any windows, and you are made to live in that. Suppose there's a house, there are 4 rooms. Each room is not having any door, any windows, anything. And there are 4 members in the family. If I keep each member of the family in one room, will they be able to meet each other in any point of time? No. Why? Because I have blocked those members in different rooms. And those rooms are not having any openings like gate or window through which those members can move out and meet each other. So such a home is actually a home where you have blocked members and they are not able to meet each other. Will that family be a family? No. So similarly in case of plants also, you have seen that the plant is made up of so many cells. Each cell are joined with the help of middle lamella. Now here middle lamella, here middle lamella, similarly here also and here also. So actually you have blocked this cell from all the 4 sides with the help of middle lamella. And this middle lamella is a cementing substance. It will act like a wall and it will not let this cell to interact with another cell, to interact with another cell. And if cells are not able to interact, how will they form tissues and organ system? Then such multi-celled organism will become a waste till the time its cells are not interacting. So God knew all those trouble. Hence he created some structures called plasmodesmata. Through the cell wall, through the middle lamella, with the help of plasma membrane and endoplasmic reticulum comes plasmodesmata. Plasmodesmata are tube-like structures that join the 2 adjacent cells and helps in their cytoplasmic exchange. So the plasma membrane of 2 cells, they are going to join to form a hollow-like structure that is going to connect the 2 adjacent cells. Clear to everybody? So now if I have to draw plasmodesmata, then how I will draw? Just see this. This is the plasma membrane which I am drawing. Plasma membrane of the 2 adjacent cells, they are coming in contact. As a result, the middle lamella and the cell wall, they are traversed from the mid. Here is middle lamella, here is middle lamella. This is the cell wall, this is the middle lamella, but the plasma membrane of 2 adjacent cells, they are connected to form a tube-like structure called plasmodesmata. So now you can see plasmodesmata. What is plasmodesmata? It is actually a cytoplasmic continuation. Plasmodesmata are the structures that maintains now here material can move here and here material can easily come to the other cell. So these are maintain cytoplasmic connections in between two adjacent cells. First point. Second, they arise with the help of plasma membrane, that is by plasma word, and endoplasmic reticulum. So endoplasmic reticulum and plasma membrane, they function in such a manner to form plasmodesmata. Clear that how a plant cell looks like, what are its characteristic features, if I specifically talk about cell wall and cell membrane, right? So with this, your cell envelope for both pro and eukaryotic cell is done. Now we are moving ahead inside the cell, and now we are going to talk about its cytoplasm. So you know, in case of eukaryotic cell, cytoplasm is divided into number of compartments. That is due to the presence of membrane bound cell organelles. So now finally we are going to start about endomembrane system. That means we are entering into the next segment of the chapter where we are going to study all the cell organelles individually. But in case of eukaryotic cell, there are 4 such structures who are functionally coordinated. They are endoplasmic reticulum, Golgi bodies, vacuoles and lysosome. All these 4 cell structures, out of which 3 are organelle and 1 is inclusion body. These 4 different cell structures that divide the cytoplasm into compartment, actually all these 4, their function are intercoordinated. As a result, they form endomembrane system, and all these 4 structures are actually single membrane bound. So one thing again which is common in them is that how many membranes are present around them? Only 1. And all the 4, I'll show you that how their functions are intercoordinated. Clear? Are you ready to see that why these 4 structures are considered as endomembrane and how are their functions intercoordinated? Yes or no? So without wasting time, without any delay, let's start the concept of endomembrane system, and then we will study each organelle separately. Right? Okay. Now my dear students, suppose this is a huge cell. This is a cell. Correct? Inside a cell there is a well-defined genetic material that is called as nucleus, which is actually double membraned. Now, to the outer membrane of the nucleus is attached what? Is attached ER. So whenever you will draw nucleus, whenever you are going to draw the membranes of nucleus, you will always show what? You will always show endoplasmic reticulum. Correct? Right? Now, endoplasmic reticulum is represented by 3 structures. Those 3 structures of endoplasmic reticulum are cisterna, which are flattened sac-like interconnected units, which I have drawn like this. The number of flattened cisterna, the number of these sacs may vary between 4 to 6. Correct? Now, these cisterna, they have some specific locations on their surface that helps in the attachment of ribosomes. The ATS ribosome gets attached on the surface of cisterna. Not all on, but not on all flattened sacs, but on some it gets attached. Clear? Coming to the next part. See in the diagram. So these are cisternas. Right? On some cisterna I can draw these roughly structures called ribosomes. Apart from this, you can see I have drawn some tube-like structure. So this is known as tubules. So what are tubules? The second part of ER, which is tubule. Tubules are actually the fenestrations of cisterna. The cisterna may get infolded further to form tube-like structures. Right? So these tube-like structures are known as tubules. Second part. Third, from the cisterna as well as from the tubules may arise some spherical bodies. These spherical bodies are known as vesicles. So overall, structurally, endoplasmic reticulum is represented by 3 units: cisterna, tubules and vesicles. Right? Now, cisterna who is having ribosome attached on their membrane, they are rough. When you observe them under microscope and you see ER, you see the cisterna of ER with ribosome, then they appear to be a little bit rough. Correct? Such cisterna of ER having ribosome attached on them is known as rough endoplasmic reticulum. Correct? And those cisterna along with tubules which lack what? Which lack ribosome, they will appear smooth. So here see, these are ribosomes attached. So this part of ER having ribosome attached will appear rough, and the part of ER which is lacking ribosome will be called as smooth endoplasmic reticulum. Correct? Rough endoplasmic reticulum is responsible for protein production, whereas smooth endoplasmic reticulum is responsible for lipid synthesis and many other functions. But here I am specifically going to tell you about protein synthesized by RER and lipids synthesized by SER. Clear? Now, so there are how many types of ER? On the basis of the attachment, 2 types, RER and SER. RER means those on whom ribosomes are attached, and the cisterna lacking ribosomes are known as smooth endoplasmic reticulum. First, in the basic difference. Second, in case of RER, the main function is protein synthesis. Whereas if I talk about SER plays number of function, like first of all, lipid synthesis. Apart from lipid synthesis, SER can also help in glycogen metabolism. They also help in detoxification of drugs. The alcohol which you take, it is detoxified with the help of SER. Correct? So these are certain functions which are performed by SER, and the main function of RER is to produce proteins. So both ER, if I say, be it RER or SER, both are responsible for producing some of the other chemical which are very, very important for the cell. You know, lipid and protein, they are used for making plasma membrane and so many other structures. Now, suppose this RER produces a protein, right? The SER also produces lipid. Now, the lipid and protein which are synthesized by SER and RER, initially they are rough. They are not ready to be prepared within the cell. I am repeating once again, the proteins and the lipids which are synthesized by the two components of ER, they are initially rough, raw, they cannot be directly used. They initially need modifications. They need certain changes in them so that they can be made ready to be used either within the cell or outside the cell. So what happens? Whatever material is being synthesized by the ER comes inside the space of the ER. The space of the ER is known as luminal space. The space enclosed within the ER is known as luminal space. So whatever is synthesized, whether it is protein or lipid, it comes within the luminal space of ER. There, the modification begins. What happens? The modification of protein as well as lipid, called as glycosylation and glycosidation, begins in the luminal space. However, ER is unable to complete that process. So, now ER has produced protein, begin the modification, but it is not able to completely modify the two components, that is lipid and protein. For that, it needs the help of Golgi bodies. Now come the second component of endomembrane, Golgi. Golgi bodies, my dear student. Now this lipid or protein, partially modified but not completely. For complete modification, it need to move out to another cell organelle present near the ER itself, represented by again flattened sac-like interconnected units called cisterna. Now, so what will happen? How these proteins will go to the Golgi body? Problem. So here comes the solution, that is the third part of ER, vesicles. So now, vesicles from cisterna and some vesicles from tubules. Tubules will carry lipid and vesicle arising from cisterna will carry protein. These vesicles are acting like scooters. Like if you want to move from one place to another, what do you use? Your scooter, now use your bicycle, or you use your car. So they are actually vehicles which carry one person from one place to another. So similarly, these vesicles are actually the vehicles inside the cell which carry proteins and lipid from one cell organelle to another. So ER is going to make a vesicle. It is going to do the packaging of the lipid and protein inside those vesicles. Then vesicles will get detached. They will come in the cytoplasm and they are ready to get transported. Now, these vesicles which are now in the cytoplasm, they will get transported and they will reach to the first cisterna of Golgi body called as cis or forming phase. So this vesicle will go and attach to the first phase of the Golgi body called as cis phase. Now, once the vesicle will attach, it is going to deliver all the chemical which is there inside it, inside the cis phase, and then it will get detached. Now, cis phase of Golgi body has received the partly modified chemicals from ER. So, ER has functioned. ER ने बना दिया protein and lipid, but for modification it has sent to Golgi. So the function of Golgi body is to modify the proteins and lipid both it received from ER. So can you see, half work is done by ER and remaining half work is going to be done by Golgi. So they both are functionally coordinated. They both are functionally interdependent, right? So the modification, glycosylation as well as glycosidation will begin. In the cis phase, also called as forming phase, modification will be done. Some modification by the cis phase, then it will transfer to the next cisterna, then next cisterna. Finally, the chemical will be delivered to the last cisterna of the Golgi body, known as trans or maturing phase. Clear? Now, this trans or maturing phase will receive the chemicals, either lipid and protein, and will completely modify them. Now, the proteins and lipid present in the trans phase of the Golgi are ready to be utilized, either inside the cell or outside. So again, for that they need transportation. So now the trans phase is going to give rise to another vesicles. So vesicles are also the part of Golgi body. Now these vesicles will receive the chemical, the completely modified proteins and lipids. That means the function of Golgi body is modification, formation of vesicle, packaging of the chemical in the vesicle, then transportation. Now these vesicles will detach and they will come in the cytoplasm. These vesicles which arise from the Golgi body, my dear students, they carry completely modified lipids and proteins. Right? Now, these vesicles which have completely modified lipid and protein, these, if the protein within the vesicle is required outside the cell, then through the plasma membrane by the method of exocytosis, the vesicle will be delivered outside. But if it is required inside, then these vesicles will not move out but will remain in the intracellular space. Suppose if the vesicle contains such proteins which can later form digestive enzymes like amylase, carbohydrates, nuclease, etc., then these proteins will form those hydrolytic enzymes. And you know what is these hydrolytic enzymes going to do? They will help in digestion. So such vesicle carrying proteins capable of forming hydrolytic enzyme can directly behave as lysosome. So lysosome is the cell organelle that arises due to the activity of Golgi body. Proteins produced by ER, modified by Golgi. Now those proteins, if they are ready to make hydrolytic enzymes, then vesicles coming from Golgi body can directly behave as lysosome. So can you see? The third organelle is also coordinated with the first two. ER, production. The function of ER is production. Function of Golgi body is modification. Function of lysosome arises how? Because it is actually made by Golgi body. Function of lysosome is digestion. For that it needs enzyme. For making enzyme it needs protein. And protein are coming from Golgi body. Clear? So can you see that how the three cell organelles are intercoordinated? Suppose this vesicle forms the lysosome. So what lysosome is going to do? It is suppose some foreign particle is moving outside the cell. So with the help of endocytosis, this foreign particle will be brought inside. Lysosome will engulf the foreign particle. Right? A pathogen, suppose it is a pathogen. So with the help of endocytosis, that pathogen will be called inside the cell. And then it will be made to enter inside the lysosome. And lysosome is having hydrolytic enzymes. Those hydrolytic enzymes will digest whatever it can digest. Some undigested part temporarily will go and get stored in vacuoles. So here comes the role of the fourth part. Vacuoles are simply inclusion bodies. They are just the storage bodies which temporarily store the excretory and the waste products of the cell temporarily, and then they also deliver outside the cell. So simply you can say vacuoles are like dustbins of the cell. So can you see how ER, Golgi, lyso and vacuole are coordinated. ER is production. This is the factory. Okay, this is the production house. Then production is not ready to be used. So it will be then supplied to Golgi body by make it ready, modify it. Once modification is done, then vesicles will come which may either move out of the cell as per the requirement. If they remain inside the cell and contains proteins that can make hydrolytic enzymes, the same vesicle from the Golgi can behave as lysosome. So lysosome came into the picture. Lysosome, digestion, some undigested particle will go and get stored in the vacuole temporarily. So this is how vacuole came into existence in endomembrane system. So endomembrane means those single membrane bound structures which are functionally interdependent. And I hope that how these four are interdependent is clear to each and every student. With this, concept of endomembrane is done, but we also have to understand the structure of each cell organelle. ER, I have already told you that it is single membrane bound, always found attached to the outer membrane of nucleus. Structurally, it is made up of cisterna, tubule and vesicles. Functionally, it can be of two types, RER and SER, depending upon whether ribosomes are attached on the cisterna or not. So this much you have to do for ER. Now, coming next is Golgi bodies. Now, my dear students, what is the role of Golgi body? Modification. What modification? Glycosylation and glycosidation of proteins and lipid which it receives from ER. So the main function of Golgi body is receiving the chemicals from ER, modifying them, packaging them in the vesicle and then transporting them. It may also give rise to lysosomes. Correct? Okay. Now, structurally, if I talk about Golgi bodies, these are single membrane bound cell organelle which are structurally similar to SER. How? Because they also contain three parts, that is cisterna, tubules and vesicles. So Golgi body also structurally consists of three parts and they are present near the nucleus in a concentric manner. This is how they are present and they are structurally represented by three parts, cisterna, tubules and vesicle. So they are arranged near the nucleus in concentric manner. It was discovered by Camillo Golgi. So the scientist in his own name kept the name of the cell organelle as Golgi body. Camillo Golgi, Golgi body. They are found in all eukaryotic cells except sieve tubes, mature sieve tubes, mature RBC. They tend to lose Golgi body on maturity. And that is the case with other cell organelles as well. Usually, in case of plant cells, Golgi bodies are known as dictyosomes. So these are certain points which you have to remember for Golgi body. Now if I talk about the cisterna, then there are two distinct cisternas. However, Golgi body may be represented by number of flattened cisternas, maybe 4 to 8. Right? But the first cisterna that receives the vesicle from ER is cis or forming phase. It is the one that receives the vesicle from ER. And the last phase from where the final vesicle arises containing completely modified chemicals is known as trans or maturing phase. Clear? So there are two distinct phases in case of cisterna depending upon their role. The one that receives the vesicle and the one which delivers final vesicle containing completely modified chemicals, cis and trans, forming or maturing. So it is very important to remember and learn. NCERT will surely ask question from this. Clear? Now moving ahead, functions I have already discussed, but here I am going to write few for you. Like number one, the main and the most important function of Golgi body is what? Modification, packaging, secretion and transportation of completely modified lipids and proteins in vesicles. Correct? Apart from this, they help in the formation of lysosome. So they help in the packaging of lysosome. Third, what modification do they do? So they help in glycosylation and glycosidation. Glycosylation and glycosidation of lipids and proteins. Right? Fourth, you know what is sperm? Sperm is the male gamete. So sperm is structurally represented by three parts. Right? The tail, the neck and head. Head is having on the tip of the head is characterized by some hydrolytic enzymes called acrosome, whose function is to dissolve the egg membrane. So this acrosome, which is rich in hydrolytic enzymes, this acrosome of sperm head is produced by Golgi body. So it makes the acrosome of sperm head, and the enzyme is hyaluronidase. Hyaluronidase is the digesting enzyme which is produced by the packaging of proteins in Golgi. So these are certain roles of Golgi body which you need to remember. I hope till here all the things are absolutely clear and crystal in your mind. Now, moving ahead, the third structure is of endomembrane system is lysosomes. Now we are going to start the third cell organelle which come in the sequence of endomembrane system, that is lysosome. Now my dear students, as I've already told you, lysosomes are also single membrane bound cell organelle that arises due to the packaging in Golgi body. First point. Second, their main role is to perform digestion because they contain different types of hydrolytic enzymes. So first point is the membrane bound vesicles that arises due to the packaging of Golgi body. I hope it is clear. I've explained you with the help of some concepts. Second, they have lysosomes. The lysosomes, they have enzymes called hydrolytic enzymes that helps in digestion. So the main function becomes digestion. Now my dear students, these enzymes, these hydrolytic enzymes, they're active only at acidic pH. So pH which is less than 3 or 4. So if you want to make these enzymes active, you want to keep these enzymes active, then inside the lysosome compared to cytoplasm, pH has to be maintained less. So suppose this is a cell, this is the lysosome, right? Now the pH inside the cytoplasm should be comparatively higher. That means proton concentration in cytoplasm should be less as compared to lysosome. So in order to make acidic pH inside the lysosome, protons, they move from the adjacent cytoplasm by spending energy. So it is an active process. Protons, they move inside, and thus the pH inside the lysosome falls in order to maintain acidic pH, so that the enzymes can be made to remain active. Clear? So enzymes, they are only active at acidic pH, and this is done actively by pumping by pumping protons from the adjacent cytoplasm. Clear? Yes or no? So this is very important point. You should remember. I hope now you will remember. Now, moving ahead to the next point, my dear students, lysosomes, which were discovered by Christian T. Diouf. He told one very interesting point for lysosomes, that is, they show polymorphism. What lysosome they exhibit? Lysosomes are the single membrane bound cell organelle that exhibit polymorphism. Polymorphism means this lysosome can exhibit in more than one form. And those four forms in which lysosome exhibit are primary lysosome, secondary lysosome, also known as heterophagosome, followed by tertiary lysosome and quaternary lysosome. Right? Now, since childhood we have been hearing that lysosomes are the suicidal bag. Absolutely correct. Yes, lysosomes are suicidal bag, but out of the four types of lysosome, only quaternary, also known as autophagic lysosome, is actually suicidal. Why? Because this form of lysosome has the capacity to burst and to release all the hydrolytic enzyme in their nearby cytoplasm, which can lead even to the death of the cell. Hence, the quaternary lysosome, the autophagic lysosome, is known as suicidal as it can burst and can release the hydrolytic enzyme outside in the cell, thus can digest the cell itself. Clear? So these are the points which you have to remember for lysosome. Moving ahead to the fourth part, the inclusion body of the cell called vacuole, which is also the part of endomembrane system. You know, in animal cells, vacuoles are very small and even they may be absent. But if I talk about plant cells, then 90% of the total cell space is occupied by vacuole. 1 point. Second point, vacuoles, they are surrounded by single membrane structure, and that membrane is tonoplast. First point, vacuole is a membrane bound space found within the cell. Correct? Second, the single membrane is known as tonoplast. This tonoplast is actually selectively permeable in nature. Please remember this point. Mark a asterisk over this. Question will be asked on the permeability of tonoplast. So tonoplasts are selectively permeable in nature. Now, what is the main role of vacuole? So if I talk about the main role of vacuole, then vacuoles are actually inclusion bodies. Their main function is to perform storage. So they can store the waste and the excretory or secretory products of the cell. They are like temporary storage grounds. They are like dustbins of the cell. They temporarily store excretory and secretory product, as well as excess water also gets stored in the vacuole, and as and when required, they are moved out of the cell. Clear? Now, one more point you have to remember, whatever material has to be deposited inside the vacuole has to pass that membrane tonoplast, and this movement is active. So, deposition of material inside the vacuole is active. So energy खर्च करनी पड़ेगी, energy need to be spent in order to store the material inside the vacuole through tonoplast. And last point, you already know, they occupy 90% of the total space of the plant cell. So with this, endomembrane system is finally over. Now we are moving ahead towards the next set of cell organelle, that is mitochondria and then plastids. So let's start with the next, that is your mitochondria. What are mitochondria? Abhi tak, till now you have read about 4 structures of the cell which are single membrane bound. But now you are going to see those set of cell organelles which are double membrane bound, starting with mitochondria. So mitochondria are double membrane bound cell organelles which are somewhat sausage shape or cylindrical in shape, and you need to very clearly stain them with specific dyes if you really want to see them under the microscope. Right? These are double membrane bound. Chalo, let's read the point. So first of all, mitochondria should be specifically stained, as I told you. Otherwise, they are not easily visible under the microscope. Right? Very important. Second, they are double membrane bound cell organelle, somewhat sausage or cylindrical in shape. 3 points clear? Fourth, now my dear students, the number and distribution of mitochondria varies from cell to cell. Their presence also, it is not as such that in all eukaryotic cell mitochondria will be found. They might be absent, like in mature RBCs, in mature sieve tube cells, mitochondria, they are lost. But if they are present, then their number and distribution varies from cell to cell as per the energy requirement. If any cell is requiring high amount of sorry. So if any cell is requiring high amount of energy, it is going to have more number of mitochondria. And if any cell is requiring less amount of energy, then in that case, number of mitochondria in that cell will be restricted. Like if I talk about plant and animal cells. So plant cells, they have lesser number of mitochondria because energy requirement in them is quite less. But if I talk about animal cells, then animal cells, we are moving, we are eating, we are talking, sleeping, so many functions we are doing. So all those functions require energy. So in animal cell as compared to plant cell, number of mitochondria are more. So here you also get one more difference between plant and animal cell. Do mention in the chart. I forgot to mention over there. So please do mention this difference as well as in the chart on the basis of the number and distribution of mitochondria. Now moving ahead. So in terms of the number of mitochondria in cell is variable, and depending upon the activity, then depending upon the physiological activities which that cell performs. Correct? Now, this is just the introduction part. One more and very important introduction I'm going to give you about mitochondria that you can see is written in green color. Mitochondria are called as semi-autonomous cell organelle. Now, what is semi-autonomous and why are mitochondria called as semi-autonomous? For this, you have to just remember or just listen to me 2 minutes. Extra from NCERT may not be asked in NEET, but still you need to listen to me just to correlate. So there was one theory given or hypothesis given called endosymbiotic hypothesis. Endo means within, symbiotic means mutual. So my dear students, there was one theory given by some scientist who believed that mitochondria as well as chloroplast, the same theory is applicable in chloroplast as well. So here I am telling you, but I am not going to repeat it over there. Now, some scientists believed that in mitochondria and chloroplast, they were actually some prokaryotic cell. Initially, mitochondria and chloroplast were considered to be prokaryotic cell which in search of shelter entered inside a bigger eukaryotic cell. So eukaryotic cell welcomed that prokaryotic cell but asked this prokaryotic cell to provide some benefit. so that there can be a mutualistic and symbiotic association. And if symbiotic association is maintained, then prokaryotic cell can live for longer time. So these prokaryotic cell, they thought that it's okay, we are getting the shelter, why not to give some benefit to the eukaryotic cell? So these cells, some of them, they started providing energy, which is now called as mitochondria, and some prokaryotic cell started performing photosynthesis, now called as chloroplast. Therefore, this concept of prokaryotic entering inside the eukaryotic, making a symbiotic association, giving some benefit to the eukaryotic cell is endosymbiosis. Right? Now, why people are after mitochondria and chloroplast? Why not to talk about lysosome and ER, that they might also have some prokaryotic connection? Because when the structure of mitochondria and chloroplast was studied in detail, it was found that these two cell organelles shows very high similarity with prokaryotic cells. Like, they can divide independently of the cell by fission method. They have their own ribosomes, which are 70S type, similar to prokaryote. Then they have their own genetic material. Apart from nucleus, they have extra genetic material, which is double-stranded circular single, same as prokaryote. Then these two cells, they have their own ATP synthesizing machinery apart from the cell synthesizing machinery. So there were certain points which was uniquely found in mitochondria and chloroplast. And these properties are directly related with prokaryotic cell. Hence, scientists thought that kya pata, we don't know, maybe these two mitochondria and chloroplast were actually prokaryotic cell which entered inside the eukaryotic cell, formed symbiosis, and then later on got modified within the cell, and now they are named as mitochondria and chloroplast. Clear? So what endosymbiosis say? That there are two cells. There were two cells. One prokaryotic cell and another eukaryotic cell. Right? So now what happened? This prokaryotic cell entered inside the eukaryotic cell in search of shelter. Eukaryotic cell agreed. Now inside this eukaryotic cell, the cell which entered has to also give some benefit to the eukaryotic cell, otherwise why the cell will keep it inside, right? So this eukaryotic cell, in two different cases, in some it started providing energy. In that case, it is now known as mitochondria, the powerhouse of the cell, and in some it started performing photosynthesis, now called as chloroplast. And this is the symbiotic mutualistic association. So this is getting the benefit, this is getting the benefit. Right, this was endosymbiotic. And why endosymbiotic is only applicable to these two cell organelles? Because there were many similarities that were found common in between prokaryote cell and mitochondria and chloroplast, and that is why they can independently exist as well outside the cell. Hence they are known as semi-autonomous cell organelle. And what are those properties that make these two organelles similar to prokaryote and that also helps in their independent existence? Number one, presence of their own protein synthesizing machinery in the form of 70S ribosomes. Apart from 70S ribosome, they also can multiply, they can increase their number by fission method, like you see in case of bacteria. Third, they have their own genetic material, DNA, which is double-stranded, circular, single DNA. Then for life for sustainability. Apart from proteins and DNA and apart from reproduction, we also need energy. And Golgi body, they cannot prepare their own energy. But mitochondria as well as chloroplast, they have their own ATP synthesizing machinery. So for these reasons, they were considered similar to prokaryotic cell, and that is why they can show independent existence, hence are semi-autonomous. So now let's try to understand the structure of mitochondria, which I told it is double membrane bound, outer and inner membrane, and they appear to be sausage shaped or cylindrical in shape. So this is the outer membrane of mitochondria. The inner membrane is present in the form of infoldings called cristae. Right? These finger-like projections of inner membrane is known as cristae. The space between the two outer and inner membrane is known as perimitochondrial, या intermembranous space. Right? Now, inside the inner membrane, again there's a space which is known as matrix. Now, this matrix actually contains double-stranded circular DNA, some 70S ribosomes, and some enzymes responsible for glucose breakdown. So this is what? This is 70S ribosome, and this is their DNA. And apart from this, they also have some enzymes that are responsible for the glucose breakdown. Now, towards the matrix, these finger-like projections, the inner membrane also have some specialized racket-shaped structures called oxysomes, also known as F0, F1 particle, and they are used for oxidative phosphorylation. So these are oxysomes, also known as F0, F1 particle, and they are actually responsible for ATP synthesis. They help in one of the method of ATP synthesis called oxidative phosphorylation. This is how mitochondria appears to be, and the main function of mitochondria is production of energy. That is why this is known as powerhouse of the cell. Clear? This much you have to do for mitochondria structure, why it is known as semi-autonomous, similarity to prokaryote, and its function. Further detailing of mitochondria is there in your syllabus when we talk about the chapter respiration. Now, moving ahead with the next set of cell organelles which are uniquely found in plant cells, and those organelles which are responsible for performing photosynthesis called plastids. Plastids are absent in case of animal cells, because animal cells, they do not have to perform photosynthesis. They are heterotroph, they are not autotroph. Clear? So let's talk about plastids. Plastids, similar to that of mitochondria, they are also double membrane bound cell organelles, right? They are exclusively found in plant cells as well as in those who are capable of performing photosynthesis, like some protisteans. They are completely absent in fungi, in monorans, as well as in animals. So they are uniquely found in plant cells, algal cells, and euglenoids. Right? Moving ahead. Now, plastids on the basis of their function and color can be of three types. Those three forms of plastids are interconvertible. So you can write that plastids on the basis of shape and not shape, function and color, it can be of three types. Can you name them? Very easy. Leucoplast, chromoplast and chloroplast. Leucoplast, chromoplast, and the third is chloroplast. All these three plastids are interconvertible. Right? So one by one I'm going to tell you about leuco, chloro and chromo. First let's start with leucoplast. Leucoplasts are colorless plastids. Why? Because they don't have any color imparting pigments. Second, leucoplast, the main function is storage of food. Now the main function of leucoplast is storage. And on the basis of this storage, again leucoplast can be of three types: amyloplast, eleuroplast and elioplast. Amyloplast, elioplast and eleuroplast. Amyloplast form of leucoplast stores food in the form of starch, in the form of sugar. Elioplast stores food in the form of fats or oils. And eleuroplast form of leucoplast stores food in the form of proteins. Right? Please remember, NEET definitely ask questions on this basis as well. Moving towards the second type of plastid, that is chromoplast. Chroma itself means colored. So these are colored plastids. They may be of variable colors like yellow, orange, purple, red, etc. And why so many variable colors? The only reason is presence of colored pigments like anthocyanins, xanthophylls, lycopene, etc. And these chromoplasts are richly found in flowers and fruits. Why? Because they impart color to them so that they can become more attractive, maybe for attracting insects for pollination. Clear? So, what are chromoplasts? Chromoplasts are the colored form of plastids that are responsible for imparting coloration to the flowers and fruits. Moving ahead with the third form of plastid, that is chloroplast. Chloroplasts are specifically green colored plastids because they have such pigments that make them appear green. And chloroplasts are strictly responsible for performing photosynthesis. Very important cell organelle. This cell organelle is keeping all of us alive. So it's our responsibility to study chloroplast also in detail. Now we are going to see the third form of plastid, which is chloroplast. I hope leucoplast and chromoplast are clear on the basis of their function. The third and the very important form of plastid is chloroplast. These chloroplasts are green colored plastids due to the presence of some pigments like chlorophyll A, chlorophyll B, xanthophylls and carotenoids. These pigments overall impart green coloration to the chloroplast. Now, chloroplasts are mainly specialized to perform photosynthesis. The main role of these cell organelle is to perform photosynthesis and help provide food in the entire world. These chloroplasts are double membrane bound cell organelle. So if I have to draw chloroplast, then how shall I draw it? First of all, it is also double membrane bound as all other plastids. There's one outer membrane and there's one inner membrane. So suppose this is the outer membrane followed by inner membrane. Right? Now, the space between the two membrane is known as intermembranous space. Short form I have written as IMS. Correct? Now, the space which is present within inner membrane, this region is known as stroma or ground substance. Stroma is rich in double-stranded circular single DNA as well as 70S type of ribosome. So they have DNA as well as they have ribosomes. Apart from DNA and ribosome, stroma also contains enzyme called Rubisco, the most abundant enzyme in the biosphere, which is responsible for CO2 fixation into glucose. So Rubisco enzyme is present in stroma, hence glucose production takes place in the stroma of the chloroplast. Now, apart from your DNA and ribosome along with the enzyme, stroma also contain some coin-shaped structures. Coin-shaped structures called thylakoids. Thylakoid is the third functional membrane of chloroplast. Apart from two membranes, the third functional membrane of chloroplast is thylakoid. Thylakoids are present in the form of piles, in the form of heaps, one above the other. These heaps of thylakoid is known as grana. Right? Now, granas are interconnected to each other with the help of stroma lamella or fret channels. So, they are connected with the help of stroma lamella, also known as fret channels. So this is how chloroplast appears to be. This whole structure is responsible for performing photosynthesis, which takes place in two parts. The two broad steps of photosynthesis are light reaction and dark reaction. Light reaction takes place in these thylakoids, in these grana, where light is trapped and converted into chemical form like ATP and NADPH2. And then dark reaction takes place in the stroma, where actually glucose production takes place while during CO2 fixation with the help of the enzyme Rubisco. So overall photosynthesis takes place in this cell organelle. The thylakoids, the membrane of thylakoid contains photosynthetic pigments. So packets of photosynthetic pigments are located on the thylakoidal membrane, which is responsible for imparting them green coloration. I hope the structure and function of chloroplast is clear. Details of this you will do in chapter photosynthesis. So with this, next set of cell organelle, that is plastids, are also completed. Now, moving ahead with the next cell organelle, which is uniquely found in both prokaryote and eukaryote, ribosomes. Ribosomes are discovered by scientist George Palade. He observed some granular structure, non-membrane bound structure. That is why they are found in prokaryote also. So the scientist George Palade found some granular structure, non-membrane bound structure in the cytoplasm of the cell, which he called as ribosomes. Ribosomes are richly made up of two components, that is rRNA and proteins. So please remember what ribosomes are made up of. So they are made up of rRNA and proteins. There can be two types of ribosomes. Ribosomes are overall two types. One is 70S and another is 80S. Be it 70S or 80S, both the types of ribosomes are made up of two units each. One larger unit and another smaller unit. The larger and smaller units of 70S are 50S and 30S respectively. And if I talk about 80S, then the two units are 60S and 40S respectively. Clear? Very good. Now each subunits are made up of different types of rRNA and proteins. Right? Now normally in the cytoplasm, when protein synthesis is not taking place, these units they are separately present. But however, when protein is required in the cell, then the two units, they attach on the mRNA and then they let the synthesis of proteins. To bring the two units of ribosome together at the time of protein synthesis is the role of magnesium ion. There is a specific critical concentration of magnesium ion which is required to maintain the ribosomal units. If that critical concentration in the cell is not maintained of magnesium ion, the two units will remain separated from each other. However, during the time of translation, the two units they are joined together on mRNA, and the reason of their joining is the maintenance of critical concentration of magnesium ion in the cytoplasm of the cell. So will you remember this thing? That usually when there is no protein requirement, the two units of ribosome, be it 50-30 or 60-40, they are separate. But when there is requirement of protein synthesis, the two units, they come together, they join, they complete the ribosomal units, and for that magnesium ion critical concentration is required. Clear to everybody? Right. Now these units 50S, 30S, 60S and 40S, everywhere I am using the term SSS. What this S stands for? So this S stands for Swedberg unit, also known as sedimentation coefficient. Now one thing you must be thinking, ma'am, 50 and 30 together make 80. But you are telling these are the two subunits of 70S. Ma'am, 60 and 40 together they make 100. And you are telling that these are the two units of 80S. So my dear lovely students, here you need not to add the two subunits to get the ribosomal. Actually, this number which has been given to the subunits like 50, 30, 60, 40 is actually on the basis of their density, right? Here it is based upon, it gives an indirect measure of the density of the subunits, and you need not to add them in order to bring the proper unit of ribosome. Clear? So each ribosome has two units, the larger and the smaller. I have already told you. Here S, which is Swedberg units, also called as sedimentation coefficient, is the indirect measure of density and size of the subunits. That doesn't mean that you have to add these units to make them 70 or 80. The numbers are not additive. So will you remember this thing? Very good. This much you have to do for ribosomes. Now my dear students, moving ahead towards the very next important segment of the chapter, cytoskeleton. Cytoskeleton are uniquely found in eukaryotes only. They are not found in prokaryotes, first point. Second point, cytoskeleton represents those minute filamentous proteinaceous structure present in the cytoplasm of the eukaryotic cell for maintaining motility, for maintaining shape, for providing strength to the cell. So what are cytoskeleton? These are fibrous, proteinaceous, filamentous, minute, microscopic structures. Correct? Read it. So cytoskeleton represents an elaborate network of filamentous, proteinaceous structures which are exclusively found in eukaryotes for maintaining the shape of the cell, providing motility as well as mechanical support. They are not found in prokaryotes. Clear? Now, cytoskeleton can be of three types. In eukaryotes, cytoskeleton can be of three types: microtubules, microfilaments, and intermediate filaments. So the three types are microfilament, microtubule, and intermediate filament. Majorly in this chapter we are going to focus about what are microtubules and what is the role of microtubule type of cytoskeleton. Microfilaments, about microfilaments you study in zoology in your muscular and that chapter, skeletal system chapter, where you study about muscle contraction and relaxation. There microfilament is required, but little bit hint I will give here as well. And third is intermediate filament, which are found within the nucleus to maintain the structure of the nucleus. Correct? So majorly we are going to focus about microtubule and their role in eukaryotic cell. So the first type of cytoskeleton that we are going to deal in with is microtubule. So microtubules are the cytoskeleton which are obviously proteinaceous. These microtubule, they are made up of tubulin protein. Tubulin is a special protein found in the cytoplasm of eukaryotic cell. These tubulin protein, they make the basic unit of microtubule called alpha beta heterodimer. So this is a heterodimeric unit, right? And this heterodimeric unit is actually the basic unit of microtubule, which is made up of tubulin protein. Now, number of such alpha beta heterodimers, they join together to form a strand, called as protofilaments. So first you are having tubulin protein in the cytoplasm. The tubulin protein then forms the basic unit of microtubule, which is heterodimer alpha beta. Number of such alpha beta units, they join together, they polymerize. N number of such units they join to form a strand of alpha beta heterodimeric unit called protofilament. And now 13 such protofilaments. How many? 13 such protofilament, they join in a circular manner to form a hollow, unbranched structure called microtubule. So if I observe the 13 arrangement of protofilaments from top, how will I see? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13. So every protofilament is n number of having units, right? So this is how, these are 13 total. So 13 protofilament strands, they join together in a circular manner to form hollow and unbranched structure called microtubule. So this is the first skeleton, microtubule. Now we are going to see the use of microtubule in the cell. What is the role of microtubule? Microtubule, which are made up of tubulin protein, they are responsible for maintaining the shape of the cell. Apart from maintaining shape, they also help in maintaining motility. How? Because microtubule, they give rise to cilia and flagella, right? So they give rise to cilia and flagella. I'll show you how that structure of cilia flagella is there in your syllabus. Apart from this, microtubule also help in cell division because they make the cell organelle centrosome. So microtubule in animal cells, they help in making centrosome, which helps in cell division. And apart from this, they maintain the shape of the cell. They also provide motility. I hope what is microtubule, the structure of microtubule and functions of microtubule is clear. Now, moving ahead with the remaining two, little bit information I'm going to give you about microfilaments and intermediate filaments. Okay, so if I talk about microfilament, they are made up of protein like actin and myosin. First point. Second point, since they are made up of such contractile protein, actin and myosin, they help in contraction and relaxation. So in muscles, you will find these microfilaments, which are one of the proteinaceous cytoskeleton. Apart from contraction and relaxation of muscles, microfilaments, they also help in pseudopodia formation during the movement of amoeba. Pseudopodia formation is also a type of contraction and relaxation. Then microfilaments, they also help in ingrowth of plasma membrane wherever it is required, like for example during the cytokinesis of animal cell. So wherever you find contraction and relaxation required, there is the role of microfilaments. Clear? So microfilaments, first point, they are made up of some contractile protein like actin and myosin. Second point, they help in contraction and relaxation. Right? So they can also help in pseudopodia formation. As well, they can also help in animal cell cytokinesis. So this is about microfilament. More detail you will do in zoology when you will study the chapter skeletal system. Now moving ahead towards the next microfilament, cytoskeleton structure, that is intermediate. Here only two things you have to remember. Intermediate filaments, first of all, they are made up of lamin protein. Second, they are found in the nucleus just below the inner membrane. So they are found in the nucleus below the nuclear membrane. Clear? What is their function? To maintain the structural framework of they support the membranes of the nucleus, they prevent their collapsing, they provide support and strength to the nuclear membrane. Right? So with this, all the three types of cytoskeleton are there on your screen. Now we are going to go back to microtubule and learn that how they help in maintaining motility and cell division. So for that, we need to learn the structures which arises with the help of microtubule. First, very important cell organelle that arises due to the arrangement of microtubule is centrosome. Centrosome are the cell organelles which are uniquely found in animal cells. They are absent in plant cells and their main role is to help in cell division. These are membraneless cell organelle. Centrosome as a cell organelle is represented by two perpendicularly placed microtubular organization centriole which are surrounded by some pericentriolar fluid, but there is no membrane around them. Even centrioles are non-membrane bound. Clear? So apart from ribosome, you also came to know about one more cell organelle which is membraneless, and that is centrosome. Focus. Centrosome is an organelle which is actually represented by two cylindrical, perpendicularly placed, non-membrane bound centrioles. Clear? Centrioles are surrounded by a fluid which is known as pericentriolar fluid, but not by a membrane. Next, both the centrioles, they are perpendicular to each other, and if we try to study the ultra microscopic structure of a centriole, then we will find that they are made up of microtubules, and microtubules are arranged in such a manner that it gives a cartwheel-like appearance. Clear to each and everyone? Okay, so first I am going to show you the structure, general structure of centrosome. Now what is the function? First I shall tell you. They are uniquely found in animal cells, and their function is to help in cell division. So how centrosome is represented? It is represented by two centrioles placed perpendicular to each other. Can you see? These are the two centrioles placed perpendicular to each other, surrounded by a fluid, pericentriolar fluid. So this is actually the centrosome also, and it is always found near the nucleus. To understand the structure of centrosome, actually we have to understand the structure of centriole, and this centriole is made up of 9+0 microtubular organization. So each centriole is made up of 9+0 microtubular organization. What is this 9 and what does 0 stands for? 9 means, first I'm telling you the theory, and then I will show you it with the help of a diagram. 9 means 9 sets, each of triplet microtubules present at the periphery. And 0 means that the center of the microtubular structure, centriole, center is not having any microtubule, rather in the center there is present a proteinaceous structure called hub. So in the center, no microtubule. Rather, there is a proteinaceous hub. Yes or no? Correct? Now you know what is 9, what is 0. So 9 means 9 sets of triplet. So total 27 microtubules will be arranged on the periphery. 9 3s are 27, and center there will be no microtubule, rather there will be a proteinaceous hub. So this is 9+0 microtubular organization of centriole. Now we are going to see it with the help of the diagram. Fine. So now you know what is 9+0 arrangement in case of centriole or centrosome. Now let's see with this with the help of a diagram. Now centrioles, they are non-membrane bound, but just I want to draw 9+0 arrangement. So for my convenience, I am going to draw a dotted circle so that I can properly draw 9+0 arrangement. But you remember that centriole is non-membrane bound. Center is what? 0. No microtubule. Rather, there is a proteinaceous hub. Periphery, they have 9 sets each of triplet microtubule A, B and C. So this is 1, 2, 3, 4, 5, 6, 7, then wait, 8 and 9. So these are 9 sets each of triplet on the periphery side. Now you can remove this dotted region which you have drawn. Now there's no need. The work is done of this dotted region, right? So you can remove it according to your convenience. Now you can label them as A, B, C. Each microtubule, you can label them as A, B and C from the center towards the last microtubule. Now, the center proteinaceous hub and the peripheral microtubule, first of all, they are connected with the help of radial spokes. So, overall, there will be 9 radial spokes which are responsible for connecting the peripheral microtubule with the peripheral microtubule with the center proteinaceous hub. Apart from connecting with the center, these peripheral microtubules are also connected with the help of Nexin proteinaceous structure called linkers, CA linker. That means C of the microtubule which is present behind and A of the microtubule which is present ahead. CA linker, like this. Correct? So this is the proteinaceous hub. This is your CA linker. Then this is your radial spoke. And these are the peripheral microtubules. Total 27 microtubules on the periphery and 0 microtubule in the center. This is how the structure of centriole looks like, and this is almost giving a cartwheel like appearance. Cartwheel, you know, in Hindi we call it as a rath. It's like a rath ka paiya. So it gives a cartwheel like appearance. Also it is a star shaped like structure. This is how centriole of centrosome cell organelle is made up of with the help of microtubule 9+0 arrangement. Now, these 9+0 microtubular organization in centriole, they also give rise to basal bodies, which are also microtubular with 9+0 arrangement. Basal bodies in eukaryote are the origin point for cilia and flagella. So we can say that first microtubules, they form centriole. Centriole forms basal body from where arises cilia and flagella. All these structures are microtubule. That is why we say microtubule help in maintaining the shape, motility as well as cell division. So you need not to study the structure of basal body. It is just same as like this, cartwheel appearance. Moving ahead with the next and important segment, that is to understand the structure of cilia and flagella. Cilia and flagella, both in case of eukaryotes, they are made up of tubulin protein. They arise from basal body and they are made up of microtubules. They are mainly responsible for providing motility to the cell. The microtubular organization in case of cilia and flagella is 9+2 arrangement, where 9 means 9 sets, each of doublet in the peripheral region. That means 9 twos are 18 microtubules on the periphery, whereas center is having 2 microtubules or 1 pair. So 1 pair, also called as doublet, microtubules in the center. So that means 18 microtubules in the periphery and 2 microtubules in the center. So overall, cilia or flagella, structurally they have total how many microtubules? They have 20 microtubules. Whereas in case of centrosome, you saw that they were having 27 microtubules all present on their peripheral region. Correct? Chalo. Now see about how this 9+2 arrangement is there that arise from the basal body. Now 9+2 means, first of all, cilia and flagella, they are membrane bound. Unlike that of centriole, which is non-membrane bound, cilia and flagella, they are bounded by membrane, plasma membrane. The inner space of the plasma membrane where the 9+2 arrangement will be present is known as axoneme. Now here 9+2 arrangement I am going to show, where 9 means 9 doublets on the periphery. Let's draw the doublets. 1, 2, 3, 4, 5, 6, 7, 8 and 9. Wait. And 9. So this is how the doublets are arranged on the periphery, and they are named as A, B. The inner doublet A, the outer one is B. Correct? Center is occupied by 2 microtubules, a pair of microtubule called as doublet. These one pair of microtubule, they are interconnected with the help of central bridge, as well as they are covered by central sheath. Central doublets are connected to the peripheral doublet with the help of radial spokes. So there are all again 9 radial spokes connecting doublet, central doublet to the peripheral doublet. Apart from this, the peripheral doublets are also interconnected with the help of BA linker, AB linker. How is it? See now. Microtubule of the one which is present at the back, and B microtubule which is present in the front. So it is AB linker, like this. Correct? So this is how the microtubules are connected to each other. First of all, let me add one more thing in this. The A microtubule, it give rise to arms which are made up of dynein protein. These arms, they have ATPase activity. They break down the energy to help in movement. Cilia and Flagella, their main function is to help in motility, and for that they need energy. So the microtubule A is associated with arms which are made up of dynein protein, and dynein protein in their arms provide ATPase activity which break down ATP to produce energy for movement, for motility. So you can make it like this, right? So from A microtubule, arms arises, and with the help of the arms, they are connected to the next set of microtubule B, and these are known as AB linker. Linkers are made up of nexin protein. Clear to everybody? Yes or no? So this is how the structure of cilia and flagella looks like. Total 20 microtubules, 18 on the periphery, center 2, connected with the help of bridge, protected by sheath, radial spokes join them. So yes, these are pink colored, are the radial spokes. Peripheral microtubules are also connected with the help of AB linker. A microtubule is also associated with arms. These arms are actually made up of dynein protein that helps in production of energy for motility. So this much you have to remember for the structure of cilia and flagella. Hope you have understood the role of microtubule in making the important structures of eukaryotic cell. Now, moving ahead with the last section of the chapter, where I will be teaching you about the nucleus, that is the brain of the cell. Brain of the cell, nucleus, was discovered by Robert Brown. You know, this is brain of the cell because it controls all the metabolic activities. First point. Second point, nucleus is double membrane bound like mitochondria, like chloroplast. Nucleus is also double membrane bound cell organelle. Both the membranes, outer and inner membrane of nucleus, they are discontinuous and they have pores called nuclear pores. Apart from this information, nucleus contains genetic material in the form of thin thread-like structures called chromatin fibers. Right? We'll talk about chromatin fibers also in detail, but before that you should know, normally every cell has nucleus. But some cells like mature mammalian RBCs, sieve tube cells on maturity, they lose their nucleus just for some specific reasons, just for specific functions like RBC to carry maximum oxygen and sieve tube cells for carrying food. Usually there is one nucleus per cell, but in some like paramecium it is binucleate. They have two nucleus, one macronucleus, one micronucleus having distinct functions. Then your striated mussels, they are multinucleate, but usually one nucleus per cell. However, the number may vary from 2 per cell to many per cell. And some cells can also be devoid of nucleus as per the requirement. Clear? So this is just the hint and the introduction of nucleus. Who discovered nucleus? Robert Brown. What is the role of the nucleus? It is the brain of the cell. How many membrane bound? 2 membrane. Both are discontinuous. Usually found in all cells, but some exceptions do prevail. Usually it is 1 per cell, but in some cases it can be many or more than 1, maybe 2 or many. Right? So now with this little bit introduction about the nucleus, let's directly jump into its structure. So here I am going to draw the structure of nucleus and then we are going to study all its part. First structure and then the parts. So as I told you, nucleus is double membrane bound, outer and inner membrane. Both the membranes are discontinuous. So first let's draw. So can you see I am drawing the discontinuous membranes. This is the inner membrane and this is the outer membrane. Inner membrane is more selectively permeable as compared to outer membrane. Why? Because inner membrane has more amount of proteins as compared to outer membrane. So this is outer membrane and this one is inner membrane. Being discontinuous, they have some openings in the mid called as nuclear pores. My dear students, these nuclear pores, they help in to and fro movement of material in and out of the nucleus, between the nucleus and the cytoplasm. So proteins synthesized inside the nucleus can move out, and some proteins which are also synthesized in the cytoplasm, if they are required inside the nucleus, can also enter in. So these nuclear pores are acting like a gate to allow the to and fro movement of material. Clear? Very good. Now these two membrane, they encloses the fluid inside. This fluid inside is known as nucleoplasm. Now, nucleoplasm can be differentiated into 3 parts. That is the nuclear lamina, followed by the genetic material which is chromatin fibers, and followed by number 3, that is your nucleolus. One by one, I will talk about all the 3 parts of the nucleus within the nucleoplasm. Nuclear lamina, chromatin fibers and nucleolus. First talking about nuclear lamina. So nuclear lamina made up of intermediate filaments. As I told you, intermediate filaments are lamin protein made cytoskeletons that are responsible for maintaining the shape and structure of the membranes of the nucleus. So here on the peripheral side, just below the inner membrane. On the peripheral side, just below the inner membrane is present nuclear lamina made up of intermediate filament. So the first part of nucleoplasm is nuclear lamina, which is present towards the periphery just below the inner membrane for maintaining the structure of the nuclear membrane. It is made up of one of the cytoskeleton nuclear intermediate filaments. Now talking about the next and very important segment of nucleoplasm, which is chromatin fibers. When the cell is in interphase, when the cells are at interphase or even in resting phase, then the genetic material is present in the form of thin thread-like structures called chromatin fibers, whose ends are connected to the inner membrane. Number of chromatin fibers varies from species to species. Like for example, in a human cell, the total number of chromatin fibers will be 46 if the cell is diploid. So what I have told you, if the cell is present in interphase or is non-dividing, is in resting phase, then the genetic material is present in the form of thin thread-like structures whose ends are connected to the inner membrane, called as chromatin fibers. Chromatin fibers, they take the stain color very densely. Hence the name is given chromatin. Chroma means color. So when you stain the nucleus, the chromatin fibers, they accept the stain color very densely and they become very clearly visible under the microscope. So what are chromatin fibers? Chromatin fibers are the thin thread-like structures that carry the genetic material, and the number of chromatin fibers, they vary from species to species. Ends are connected to the nuclear membrane. Apart from chromatin fibers, nucleolus. Nucleolus are the darkly stained, non-membrane bound region present in the nucleus. The number of nucleolus may vary between 1 to many depending upon the requirement of rRNA and proteins. So what is nucleolus? Nucleolus are darkly stained, non-membrane bound structure present within the nucleoplasm of the nucleus and are responsible for the synthesis of RNA and proteins. Now, the number of nucleolus per nucleus may vary from 1 to many. If more amount of protein is required, if more amount of RNA is required, the number of nucleolus in the cell will be high. If less amount is required, then automatically the number of nucleolus will go down. So this is how the nucleus appears to be. That means nucleus structurally is having how many parts? Nucleus structurally can be divided into two parts, as the membrane system, and membrane encloses the fluid called nucleoplasm. It's a double membrane bound structure, so there are two membranes, outer and inner. Right? Now, outer membrane. First point, usually from the outer membrane, cell organelle ER is found associated. So from outer membrane, you can also draw cell organelle ER. This I have already told you. Sometimes from the outer membrane, ATS ribosomes are also found attached. So due to the presence of these ribosomes, outer membrane may also appear rough. So outer membrane, first point, discontinuous, you know this, which is common for inner membrane also, may be found attached with ATS ribosome, so it will appear rough. then also found attached with ER, and they're comparatively more permeable than inner membrane. Inner membrane, it is less permeable, or you can say more selectively permeable. First point, again it is also discontinuous. Pores bane hue hain, that now you know. Clear? So this is about the membrane system which you have to remember. Coming to nucleoplasm, nucleoplasm as I told is divided into three parts. Lamina, that is made up of intermediate filaments, one of the cytoskeleton. Talking about chromatin fibers are the thin thread-like structures carrying the genetic material which accept the stain color very densely. Clear? These chromatin fibers are made up of DNA, RNA, histone as well as non-histone proteins. Richly they are made up of DNA. They also have RNA. Their packaging is done with the help of both histone and non-histone proteins. Right? Now, nucleolus, they are the darkly stained non-membrane bound structure responsible for active in protein and RNA synthesis. Number varies depending upon the requirement of protein. So this is how the nucleus appears to be. Now we are going to talk about chromatin fiber in little bit more detail, and then we will study the structure of these genetic material carrying structure. Okay, so now coming to structure of chromosome, that means we are talking about the chromatin fibers. So first of all, I told you when the cell is present in interphase stage, so at that time the genetic material is represented by thin thread-like structures called chromatin fibers. First point, the number of chromatin fiber varies from species to species. These chromatin fibers, they contain DNA, RNA, histones as well as non-histone proteins. Correct? Now, these during the time of cell division, when the cell escape interphase and enters into M phase, that is the dividing phase, these thin thread-like chromatin fiber, they condense. The ends of the chromatin fiber detach from the inner membrane. They start condensing to form thick rod-like structures called chromatid. This is the point of attachment. When one arm is attached, when one complete arm is attached to the centromere, it is called as chromatid. Now, if two are attached, then it is known as chromosome. So, I can mention you one more thing, that since during interphase, the DNA undergoes replication. So, this thin thread-like chromatin fiber will make its one more copy and will appear to be like this, which will undergo condensation during M phase to form thick rod-like short chromosomes. Right? So one chromosome consists actually of two chromatids. So you can say that one chromosome consists of two chromatids. Right? Now we are going to study the structure of this chromosome, also called as chromatids. Are you all excited? Have you all understood till here? Okay. Now, moving ahead with the structure of chromatid. So, structure of chromatid or chromosome. You should know that one chromosome is represented by two chromatids. So, here I am just going to portray the structure of a chromatid, and one more chromatid, if is attached to the centromere, it makes the chromosome. So, if I say structure of chromatid or chromosome, you should know it's almost one and the same thing. So, here I am going to draw just one complete arm, that is chromatid. So, The chromosome or chromatid is represented first by two halves of the arm. The total arm is divided into two halves, the upper arm and the lower arm. This is upper arm and then this is the lower arm. Right? The upper and the lower arm, they are joined to each other with the help of primary constriction. Can you see? This is the upper arm, which is known as P arm. And this is the lower arm, which is known as Q arm. Now, can you see the upper and lower Q arm? They are joined at a point where some constriction is there. This is known as primary constriction, also called as centromere. So the two halves of the arm, they are joined together at centromere. Around the centromere, they are found some disc-shaped trilamellar proteinaceous structure called kinetochore. So the proteinaceous disc-shaped structures, kinetochore, that helps in the attachment of spindle fibers. Spindle fibers के attachment में this structure helps during the time of cell division. Correct? Now these arms, they enclose the genetic material. So here you can mention DNA. Correct? In some chromosomes, apart from primary constriction, there is one more constriction called secondary constriction. Right? So in some, not in all, in some chromosomes, apart from primary constriction, they have one more constriction called as secondary constriction, also known as NOR. NOR stands for nucleolar organizing region. Nucleolar organizing region. This NOR is responsible for the formation of the nucleolus within the cell, within the nucleus of the cell. So this is secondary constriction. Now, secondary constriction, they have their own important function, like one I have already told you related to nucleolus, and second, they can also act as selectable markers that helps in biotechnology. Now, the genetic material which is present beyond secondary constriction, this portion, it is known as satellite DNA because it is extra, na? This genetic material is extra. So it is known as satellite DNA, and such a chromosome having satellite DNA is known as satellite chromosome. Right? So this is how the structure of a chromatid appears to be. Now from here you can attach one more arm, P and Q, to make the complete chromosome. So one chromosome is actually represented by two halves of the arm, upper and lower arm, which are joined together with the help of primary constriction called centromere. Now centromere is surrounded by kinetochore that helps in spindle attachment. They contain genetic material called DNA inside it. In some chromosomes, apart from primary constriction, they will also have secondary constriction, also called as NOR, and the genetic material present beyond the secondary constriction is known as satellite DNA. Now the ends are sealed by, very important, ends of each arm, they are sealed with the help of telomere, having telomere. Very important. So ends of each arm, finally they are sealed with the help of telomeres. This is how the structure appears to be. Now, depending upon the position of centromere, there can be four types of chromosome: metacentric, submetacentric, acrocentric, and telocentric. So depending upon the Position of centromere. We have 4 types of arrangement which we can divide in 2 categories as isobrachial and heterobrachial. Iso means same. So under this isobrachial comes your metacentric chromosome, when the centromere is exactly in the center like over here, such that the length of both P and Q arm becomes exactly similar. Such chromosome appears to be V in shape. Then now centromere start moving away from the center more towards the P arm, then surely the length of one arm is going to increase and the length of another arm is going to decrease. So now all the 3 remaining types of arrangement will come under heterobrachial, and they are submetacentric, acrocentric and telocentric. Submetacentric means centromere slightly move away from the center, such that there is a slight difference in the length of P and Q arm. In submetacentric, the chromosome appears to be L in shape. Acrocentric, और दूर जा रहा है. Centromere is moving more away from the center, creating more difference in between the length of P and Q. Hence, the shape becomes J. And telocentric means the centromere has finally reached to the tip of one of the arm, such that one arm is completely lost and chromosome is only represented by the other half arm. And in that case, the shape becomes I. So I hope heterobrachial, isobrachial, meta, submeta, acro and telo is clear along with their shape. Submeta means L shape. Acro means J shape. Telo means I shape. And metacentric means V shape. Correct? So with this, finally the cell chapter is over. I hope you all have understood the cell chapter very clearly, and mainly I have covered NCERT in this one short video. Only NCERT important points have been covered, which will be very important for you to crack your NEET examination that is just 10 or 12 days ahead. So please students, pay attention to this, and soon I am going to keep a live session where I am going to help you in solving questions as much as possible. So in that live session, you will get an opportunity to solve number of questions back to back which have been asked in previous NEET examination or other competitive exams. In that class, whole 11th and 12th syllabus will be revised in one shot. Hardly 3 or 4 hour class will be there, and your whole 11th and 12th syllabus will come on your tips. Majorly, I am going to cover the only important topics from each chapter. Not whole chapter, but only important topics from each chapter. That is why that session on 7th July becomes very, very important for you. So please, if you are listening to this video, if you're hearing to this video, then do spread this message as much as possible, that join that live session on 7th July for your NEET examination. Just 1 week before NEET, whole biology, whole botany of 11th and 12th will be revised. Clear? Now, moving ahead, the last segment as usual is question-based segment. So some 1 or 2 questions I have. Quickly we'll solve and we'll end the session. Which cell organelle is involved in packaging of lysosome? That means, in simple, the question is asking that which cell organelle helps in lysosome formation? Options: ER, Golgi, Nucleus, Ribo. So the correct answer is B, Golgi body. Right? Very good. Moving ahead. Hydrolytic enzymes are active at which pH? In case of lysosomes, you know, lysosomes, they have 40 different types of hydrolytic enzymes that help in digestion of foreign particles, and these enzymes are only active at acidic pH. And to maintain acidic pH, there is uptake of hydrogen ion actively from the cytoplasm inside the lysosome. So correct answer will become B. And finally, with this, your chapter cell is over. Thank you for joining me. Do not forget to join the live session on 7th July. It is surely very important chapter for those students who want quick revision of all the important topics of class 11th as well as 12th. The revision will be done in your question mode. That is again very important, because now everybody must have read everything. The thing is question solving, question practice. So here I will help you to solve as many questions as possible. So in that live session, it is going to be very interesting. I will give you the questions, ask the answers, explain them, solve them, and we'll have lot of fun. So that's going to be a very fun-loving session. Hope to see you soon on 6th July. Thank you for joining me. Keep working, keep working hard, surely. Thank you.